I mean, I'm really honored to have both James and Lyle here who are going to be presenting. I just wanted to let you know that, we've been working with James and Lyle from the inception, so almost like 3.5, 4 years now, and we've done some amazing stuff together. And, without these rock and pillars, we wouldn't be where we are, even though we're patient research, citizen research. It's about really collaborating. And today we will be bringing some of these exciting work that we've been doing. So with that, I will invite Dr. Berry to start the presentation and give us the update.
Thanks so much. I mean, I second that. It's an amazing team at Everything ALS. I mean, Indu, thank you for putting it together, and really just lots of fun. And we get to have lots of meetings where we kind of talk about interesting data. I'm going to do my best starting the slideshow. Is this the right? Are you guys seeing the correct view here?
Yes.
Okay. So Everything ALS is really doing more than any individual study. I mean, I think the way to say it is that the idea is that Everything ALS is creating a digital health technology landscape in ALS. And that's remarkable because it's sort of setting out to push the whole field forward and sort of raise all boats. So the promise of digital health technologies in ALS, I think it's maybe obvious, but maybe not. ALS causes progressive change in function. Quantifying function can help track disease progression. That's important for trials. It can also be important in clinical care to have a good sense of how things are changing, and also to help us time interventions. Subjective scales like the ALSFRS-R are really useful. I don't think that we should hear today at all that the ALSFRS-R doesn't have a place or isn't beneficial, but it's only semi-quantitative and it lacks granularity for certain kinds of measurements. So there is an opportunity to add to it, do better than it for certain uses. And measuring function in clinic can fall short. Again, can be very useful, but it can be time consuming. It doesn't always reflect real life function. So there's data showing that people do better in clinic than they do in real life, in part because we have very sort of limited tests that we do in a very sort of not real sort of artificial environment. And it quantifies a limited number of functions in a fairly limited way. We do a timed up and go test and we get a time, but nothing more than that. Measuring function with DHTs holds really more promise. It's flexible, so it can be collected passively or with brief tasks at home or even in the clinic. And sometimes data can actually be collected during a tech-assisted task. So we'll hear today about brain-computer interface and how that can be useful as assistive technology, but we can also collect data during that. It can be representative. Quantifying real-life performance is relevant automatically. It's broad. Many functions and tasks can be quantified using different modalities and can be quantified quite carefully. And it's analyzable. I think one of the concerns, 5 or 6 or 7 years ago was that we might generate a heap of data and have no way to analyze it. And that's just simply not true anymore. And you'll see some examples of that from some of the work that Everything is doing. And one of the first studies that Everything ALS started was the speech study. And it's been wildly successful. But when we want to take one step back and say, why do this? Well, as we said, ALS functional deficit depends on what region is affected, and the ALSFRS-R only provides an overview of function. So quantitative motor speech analysis can provide detailed analysis of bulbar function. And we can look at things like speaking rate, which does change in ALS, but we can also look at higher level analyses, which makes us take a step back and say, what really matters about speech? And what really matters is, can speech be understood? And so then the question became, is there a way to look at whether speech can be understood or quantify that? And listener effort really, Jordan Green has done this in a very small, bespoke way, but set the stage to be able to go forward with it. And I'll tell you why in just a minute. You'll understand why that was not simple to take from a small idea into a large implementation. And that's really what Everything ALS has done, so that we could really test this on a broad level. Listener effort really asks the question, how hard do you have to listen to understand a person who's speaking? How hard is it to understand somebody? Well, that's how we talk about speech. And that's what's very fundamentally important about speech. And so it's quantifying something very, very inherently clinically meaningful. The more dysarthria somebody has, the more you have to listen carefully to understand, the more effort you have to put into it. And we can quantify that on a scale of 0 to 100. And what we've done is have ALS expert speech and language pathologists, or SLPs, listen and rate people's speech on a scale of 0. Doesn't take me any effort. to understand them. They're very, very clear. To 100, boy, as hard as I listen, I really can't make out what's being said. So one of the things about this is that you need a system to do this. And if it's going to be done at scale, the system needs to be robust, it needs to be reliable, and it needs to be simple. And so the tech engineers through EverythingALS with Indu sort of guiding them, built a really clean, simple interface to implement this. It has a warm-up screen so that the SLPs, when they sit down to do their job, can get ready for the task, and we don't have sort of a difference because, well, that was the first thing I listened to that day, and my head wasn't in the game. And then there's a very simple visual analog scale, which you see over here, where the SLP can click on the recording, listen to it, and then very simply rate that recording and make a comment. Again, very simple, but this needs to be built. And, a lot of times in academia, this is very, very difficult to do. So I think even this is a true innovation and allowed the speech study to move forward. Now, the EverythingALS speech study has enrolled hundreds of people. At the time that we did this, it was 529 participants, and this comes from a paper that is in submission right now. Of those, we selected 105 people with ALS and 20 controls to be a part of the listener effort study. And these were people with longitudinal data and good quality data that we could put into this study. We also looked at subsets, and I'll show just a little bit of that data. What's important is that we had 3 speech and language pathologists who were listening to each of these recordings. Each participant had many sessions, and the speech and language pathologist also listened to some sessions more than once so that we could look at intra-rater reliability, inter-rater reliability, and get an average listener effort. And what we found was that listener effort, inter-rater reliability, and intra-rater, I'm showing the inter here, but intra-rater was also very high. Of course, you see some variability, but the ICCs are incredibly good. And that gave us a lot of confidence about sort of the way that this task was being done. The other thing is, just as a gut check, listener effort correlates with self-reported speech function in the ALSFRS-R. And this is really important. I think clearly you see a spread. So you can see on the x-axis, the ALSFRS-R self-report, question 1, and on the y-axis, listener effort. And if somebody rates themselves as having normal speech, most of those have very, very, very low listener effort. There are a couple that are higher. They may be outliers. There could be a variety of reasons for that. The reality is no data is going to be perfect. But what we see in 2 and 3 and 1 is 3, 2, 1 is that you see a higher and higher listener effort, just as we would expect. And also, I think making the point for why this is important is that in especially 3 and 2, you see that these are very broad categories, that speech can vary wildly when people put themselves in category 3 or category 2. Not that the people are not rating themselves correctly, it's just a very broad scale that we're asking them to fill out. The other thing is that listener effort, we found, informs us about ALS progression. So listener effort does increase for people with ALS. It does not increase for controls. And that's important. We don't think it should increase for controls. And we do think that it should increase for people with living with ALS if it's going to be a useful outcome measure or clinical measure to follow. We also then looked, so in the purple and red is people with just focusing on the people with ALS and comparing non-bulbar to bulbar. And we found, as we sort of expected, people with bulbar onset had faster progression than people with non-bulbar onset. But we took one step back and said, well, let's think about this. Some of those people who started with, say, limb onset disease continue to have limb onset disease. And when they're participating in our study, they don't think that they have any bulbar involvement. But some of those people with limb onset disease, have had bulbar involvement, have it, and the disease is spread there. So let's look at the people let's compare the people who had bulbar onset to the people who have non-bulbar onset, but now have involvement of the bulbar region. What we find is in those two groups, whether it's limb onset or bulbar onset, if the bulbar region is involved, the progression is faster, and it's very similar to one another. Those slopes are not statistically distinct. So it turns out that the non-bulbar actually divides into 2 groups, people who had limb onset or non-bulbar onset and do have bulbar involvement now, and people who don't have bulbar involvement now. And that's actually a more important distinction. So we're learning about the disease and thinking this through and being able to sort of test our hypotheses with this quantitative data. Now, all of that I talked about was a ton of work, particularly on the part of these expert SLPs. And I mean, they were incredibly dedicated to rate just thousands of these recordings. But the next thing that the EverythingALS data science team set their sights on was really building a robust machine learning predictor of listener effort. And they did just that. And you can see the factors that went into this. Whisper is a automated speech analysis program. And the confidence of that program is a big factor in what determines listener effort. But speaking rate and performance also inform this. And you can see that the R-squared is 0.83. So there's a very good correlation between the machine learning algorithm and what the SLPs say, which is quite remarkable. And I will also say that this has been tested in an external data set, did just as well, both on the listener effort side and the machine learning prediction of listener effort, which is incredibly exciting to train this on one set and then test on an external set and see this kind of robust result. Really exciting. And that data we hope will be presented at some point soon. So the other thing that's exciting about what EverythingALS is doing is that it's sharing the data. So this data can be requested on the web, and the actual speech can be provided to speech researchers to push the field forward even more. One other thing I'll say is that we know that most research in the US with people living with ALS has about 90 to 95% of participants are white. That is not the case with this EverythingALS data set. It's a much more representative population. And that's something I think to be really, for the EverythingALS team to be really proud of, but also for the rest of the ALS community to look at and say, what are the factors that made that happen and how do we replicate that? Because we want representative participation. It gives us better answers. The Radcliffe study is sort of a follow-up study to the speech study. Does include speech, but it also includes remote ECAS, FEATME with pressure sensors that tells us about gait, and home spirometry. And Lyle is sort of working with the team to lead the analysis of some of this data. And it's quite extraordinary. And I think we're going to have incredible insights coming out of this. The other thing that EverythingALS is doing is really providing DHT services for trials. So the RAPA 501 EAP program, which is NIH funded and at 10 centers, will enroll 40 participants. I think about 15 of those have been enrolled now. And those participants are really signing up for an expanded access program. It's run like a trial. So, more or less they're signing up for a trial. It's open label. They come in for an apheresis and 4 infusion visits and follow-up visits, but that's what they're enrolling for. And the DHTs that we're involved, that we're using are secondary to it. This is a different population. And what EverythingALS is doing is really not only getting the data from cell phone apps, from home spirometry, from accelerometry, but actually testing the hypothesis that this can be done in a way that participants will accept it and will be able to comply with it and give us rich data. So this is actually going incredibly well. I'm a PI of the trial and one of the active site investigators, and we get really good feedback thus far on this. So really exciting direction, and I think something that we really need in the field so that we can move from observational studies with people who are interested in DHTs to trials where people are enrolling in the trial, and we have to sort of get this data from them. So there's a lot of ongoing work, validation of the SLP-rated listener effort and the prediction model, home spirometry, the sort of accelerator lab setup to test new DHTs quickly, which is what much of today is going to be talking about, and Lyle will talk about, and a combined analysis of endpoints, and then a creative search for more data. And I'm going to finish with just a couple of slides here that come from an analysis that Indu and Ernest Frankel and I did with our colleague Elad Yom-Tov, who was at Microsoft Research at the time. And he had done research really just saying, look, there is so much data that's coming into the internet every minute, and we can use that for health research. And he demonstrated that search engine interactions could actually track searches that reflect events in real life. So this is sort of searches that reflect women going through a pregnancy, just looking at their searches over the course of 40 weeks. And it's fairly remarkable. And nothing like this had ever been done for ALS. So we asked the question, could we use interactions with the search engine and the data that's generated from that to differentiate people who have ALS from those who don't? And the answer is that we could do that. And we could do that fairly robustly, actually. So here you see what the ROC with an AUC of about 0.9. There are 3 lines there. Those 3 different colored lines actually represent looking at applying our algorithm at different times in the course of the disease for people with ALS. What we found is that it didn't make much difference. So we couldn't track changes longitudinally at this point, at least using the methodology that we used. But we could differentiate from people who didn't have ALS. And there were 4 critical variables: session duration, autocorrect, the likelihood of that query, and then whether it was a very rare query. And so there were other variables that went into this, but those were the 4 that were sort of most predictive. I think the main point that comes out of this to me is to say there's data out there that is completely untapped, and we need to be creative in how we think about it. And EverythingALS is doing that and has this flywheel turning, and I think we're all benefiting from that. So incredible team. And with that, I'm going to pass to you, Lyle, to take over.
Keep that slide on the screen for a sec. Is that good? So yeah, I'm going to be, I'm the one who talks less and slow, right? It's usually my fault that we're running behind, not someone else, right? No, but so look, I'm going to be really fast because I want to get to the fun part of today. But so every week, James and myself and Ernest Frankel have these meetings with the EverythingALS data science team, right? And it's just amazing. It's when you're a kid and you think about what it means to be a scientist, this is it, because you just get to focus on figuring stuff out. And there's all these people on the Zoom that have these diverse wisdom and the data science crew, who I think you're going to hear from some of them later today. And James mentioned the speech language pathologists who are involved in these studies. And we literally get to sit there and just figure stuff out. And it's an amazing thing. And, and also James just said, oh, this is all this stuff that's about to come. Yeah. Like, I feel like a little kid being like, what are we going to do next, guys? Like, what's coming next? What's the next thing? And it's amazing. And in a more broad sense, it's impossible not to be excited by all these crazy tech that we keep seeing every week, all these digital sensors and, things that use our cell phones and, algorithms to analyze the way people are speaking. And the potential is obvious for helping us develop drugs and take care of people living with ALS. But the problem is, every study, every piece of tech, like, how do you choose and what do you do? And right now, each individual company or lab or person who has a great idea, they're kind of stuck on their own, right? They have to compete for funding and then, maybe pay for some small one-off study with one academic institute or a clinic or something like that. And there's no standard thinking about, like, what's the ground truth? How do I know that this measure is representing what it's supposed to? There's no standards for the digital data. It's awesome that we're sharing data, but it's not the same as like sharing, say, measures in biofluids or clinical measures. There's got to be standards for what is digital data, and how do we compare these different measures? And James mentioned, we talk a lot, bulbar onset, limb onset, like there's 47 flavors of ALS, and a sensor that just measures one thing about someone is not going to be the answer. And so, how do we combine these things? How do we do it together? And then, the final part of that is that the regulatory pathways for this stuff are murky and evolving. And it's not clear when the one person with the cool idea partners with the one clinic and gets some amazing data. Like, what are the goalposts? What's the goal? What's the deliverable that says, okay, and then what comes next? And so I think that the EverythingALS, call it what you want, infrastructure community, really has the opportunity to kind of take the lead in figuring this stuff out. And this first panel today, I think it's pretty cool because we've got 3 different people. So Patrick Mercier is an academic at UCSD who's going to talk to us about some amazing stuff he's done. He founded this Center for Wearable Sensors, right? And this unbelievable tech and how it's being used and sort of the perspective of an academic in this space and what comes next. And Dr. Verma is a neurologist. He's a neuroscientist. He's been a biopharmaceutical executive with lots of companies that develop drugs for ALS and other diseases. And now he's working in venture capital. And I think, has this perspective of working with these small companies and trying to help them get to the next step. And Dr. Nadine Tatton, is from, working with CPATH. And CPATH is this nonprofit. The whole point of them is to help us develop drugs, right? And help get regulatory approval. And what's the FDA thinking about? And I think, honestly, CPATH's ideas about this space and how they can help and what their role can be are evolving too. And so each of them is going to talk a bit. I think we can hold questions till the end, but please put questions in the chat. And the reason I say till the end is I really want to hear the different perspectives on similar questions. So with that, I think Dr. Mercier is going to lead us off and talk to us about what he's doing in their center.
Sure. Hello. Can everyone hear me?
Yes.
Perfect. All right. Let me just get my slides up here. All right. And we're running a few minutes late, so I'm going to try and be as quick as I can. So my name is Patrick Mercier. I co-direct the Center for Wearable Sensors at the University of California, San Diego. And we're doing, I'm an engineer. We're doing a lot of really cool technology development, and it's really great to, discuss and collaborate with folks on the clinical side to eventually push this through translation to actually help people in real life. And that's always our objective here. So just kind of very briefly, our motivation for what we're doing. Wearables are an exciting, possibly high growth marketplace. Okay, there's a lot of really cool ideas that have been thrown out there over the last decade. In some cases, wearables make real money. Apple's wearables division makes more money than their iPad division. So this is serious business for them. But there's also some products that have failed. Google Glass, for example, was a really cool idea, but just didn't quite, make it. So what I'm showing down here is an intentionally old projection from 2015 about wearables across different market sectors, suggesting, nice, beautiful exponential growth. And in some cases, this happened for, Apple Watch type of products. But in a lot of cases, and especially looking here at medical, this hasn't quite happened, at least to the way that we would have originally envisioned it. And if I could summarize with one picture why I think that hasn't happened, I would choose this picture. All right, so this person is clearly not happy with their wearables. They're not smiling. So what's going on here? Well, number one, a lot of these devices are too big. They don't seamlessly integrate into daily life. They're hard to use and so on. We need to fix this. Number two is a lot of these devices have rather poor battery life. All right, so I have a smartwatch. I have to charge it every day, and that's fine if I'm capable of doing that and it's only one device. But as soon as I have more devices, or maybe I have a movement disorder, it makes it hard for me to do this. This is problematic, right? And last, but certainly not least, these devices have to do something useful. Okay, so I have my smartwatch, it tracks my movement, but I don't go to my doctor at the end of the day and say, hey, look, doc, I walked 10,000 steps today. What's my prognosis, right? Clinicians don't care. All right, we need to develop new sensor technologies or new algorithms to analyze existing sensor technologies to deliver actionable insight into a patient's life. And so that's really our objective in the Center for Wearable Sensors, is we want to try and address all of these issues. And it's not going to be a bunch of electrical engineers like me sitting in a room solving all of these things. It needs to be transdisciplinary research, and we've structured our center to do this. So very briefly, our center is structured around multiple core research thrusts. We research on new sensor technologies to pair that with ultra-low power bioelectronics and bioenergy harvesting. And we vertically integrate with new platform technologies, like, for example, flexible electronics. And we do pride ourselves in going a step further than this. A lot of universities and groups on the engineering side will make a cool new device, test it on the bench, write their paper and move on. We like to try and take that extra step and build a full system that we can put on human patients and actually try this out. And so the outcomes are quite interesting. One is, in many cases, we're developing devices that are generating data that has never before been measured on freely behaving human subjects. So the opportunities for data science and analytics are massive. And we're also training very uniquely qualified students who can speak across technical boundaries. Okay. So with that as an introduction, let me just very quickly run through a few example projects that we've worked on over the last few years. This was a project on a wearable device that was the first one that could monitor a physiochemical parameter and an electrophysiological parameter on a freely behaving human subject. In this case, we did real-time lactate monitoring simultaneously with ECG monitoring. Okay, so this is interesting for athletic applications. also happens to be very interesting for applications like sepsis and so on. And I'm sure we can find a lot of other interesting applications for this type of technology. So building on this, we went ahead and did what we call a dual fluid sensing system. It's a little temporary tattoo, just like a kid's temporary tattoo. You put it on your body, you dab it with a bit of water, peel off the paper, and it's kind of staying on there. And what's cool about this particular technology is we do electronic delivery of a drug, pilocarpine. So there's no needles or anything like this. It gets electrically injected. And what this drug does is causes sweating to percolate to the surface in that local area. We can then do electrochemical analysis on that sweat and monitor, in this case, alcohol consumption. And at the same time, if we're going to run a current through the body, which is a small and safe amount of current, doesn't hurt, we can also pull glucose molecules out of interstitial fluid and do analysis at the surface of the body. So in this case, we could do alcohol and glucose sensing, both in real time. We had to get university approval to buy beer and give it to students, but we did that. For some reason, experiments always happen on Friday afternoons. Not sure why, but anyways, we ate, we drank, we did science, and we had some fun. And we got pretty great results, nice linear correlations with our clinical gold standards. Now, since then, we moved on to microneedle technology. This is minimally invasive microneedles. They don't hurt at all when you put them on. But what's nice about these is they give you direct access to interstitial fluid, and that allows us to do multiplex sensing of multiple different analytes, all in real time, all with very high accuracy relative to clinical gold standards. So we're really excited about this type of technology. Now, moving beyond the skin, we've also developed things like smart ingestible devices. These are pills that you can swallow. They go into your stomach and your GI tract, and they can monitor physiochemical parameters in real time in that vicinity. So this is really exciting and impactful because there's really no other way to do this, right? The only way to get analytical information about the gut is to do an endoscopy, which is a very invasive procedure. And in this case, you just swallow a pill and we get real-time information as opposed to one spot sample. In addition, this technology actually doesn't even have a battery. What we've done is we've built a glucose biofuel cell that harvests energy from glucose naturally present in the GI tract to power the entire thing. In addition, we have some interesting work on gas sensing. We've shown that we can do really, really sensitive detection of, for example, SARS-CoV-2 in air. And do this with systems that have extremely low power consumption. So we could build these into wearable devices. We could, put these on the wall inside of offices or something like this. And they're so sensitive that if someone walks in with some sort of viral infection and we're sensitive to that particular virus, we'll be able to detect it almost right away. And then the last thing I wanted to end on is, a lot of our research is focused on externally wearable devices. But we also work on neurotech. I saw there's a neurotech session later today. Just wanted to say just a few words about some of our work on neurotechnology. One of the big problems with brain-machine interfaces, neurorecording, and so on is channel density. It's really hard to fit a lot of channels in a small area. And there's physical electrode constraints at play here. But there's also electronics constraints. And so we've done a lot of work on building really robust, super high density neural recording front ends. In fact, our front end has the lowest area per channel amongst any work published in the world, and still has good performance in terms of noise and bandwidth and so on. And in addition to that, we've also done a lot of work on building much more efficient neural stimulators, such that we can, stimulate over a larger number of channels or deliver more current to the same number of channels while maintaining compliance with regulations. So that's, I think, all I wanted to say for now. Looking forward to the discussion later.
Thank you, Patrick. And I think we'll move on to Ajay, Dr. Verma's talk now. And I think we will then have questions, as I said, at the end.
Great. Can you see my screen? Can you guys see my slides?
Yes.
Awesome. Great. So I'm AJ Verma. I'm a neurologist, and I'm currently running a venture studio, a little different than venture capital. More on that another time. But I wanted to address some of the questions that Lyle had posed to me for this session. And specifically, I was focusing on, there's so much happening. It's so exciting. It's overwhelming, actually. But how do we practically scale, all this technological revolution to help patients, to help doctors actually help patients? And, what are the key considerations that we need for that? And how can we actually transform neurological assessments with that? So sort of thinking about that. And obviously, there's lots of limitations. So there's no, we have plenty of need for help. Traditional assessments are great, but there is variability in that. And the digital transformation does offer opportunities to fix some of that. Limited access is a key problem for patients to get access, come into the specialized centers, see neurologists and so on. So digital technology is very well suited for that. The key question for me is how do you, what Lyle was asking earlier, how do you make objective measures that are believable? Neurologists are pretty persnickety, they like to do their exams very carefully and always question, did they do it this way? Did they do it that way? Is this really reporting what I think it's reporting? And so in that sense, I've actually been focused on using video analysis as a bridge. Much of what we do in an examination of a patient is observation. And even if you don't lay hands on a patient, you can amass so much information just by watching someone do certain maneuvers and so on. And sort of applying video analysis and overlaying it with some of the sensors may actually help standardize and create a bridge between a neurological exam and a digital exam. And I wanted to just share a couple of examples. I had the pleasure of meeting Dr. Shin earlier at the ADPD meeting this year, and he gave an amazing presentation. I've been delving into the work from his hospital, the Seoul National University Hospital, where they've actually been doing standardized video recordings as a routine procedure for their UPDRS assessments. I know this is Parkinson's, but it's relevant. And their gait assessments, particularly for deep brain stimulator implantation. And they just collected this from 2004. And now as new technologies are coming online, they're able to feed these videos and reanalyze them. And they've actually made some remarkable progress in developing a whole new digital gait tome. And the video analytics, the computer vision that they've been able to overlay onto this very standardized measurements can give you a great insight into what really matters, which features of the gait might be the most important for predicting progression, for predicting treatment response, rate of progression, things like that. So, this is just one example, but what I liked about it was they took a simple procedure, they standardize it, same hallway, it's simple, they know the distance, the various simple maneuvers, turning around, walking back, posture, arm swings, and then just reams of data that's available there now for reanalysis. And now they've been overlaying it with, the various gait right mats and overlaying other sensors, but there's a golden truth there because these were actually done as a clinical exam. Decisions were made based on that and were recorded. And now going forward, the same standardized platform that can be easily set up in any hospital or any clinic serves as a benchmark for the new digital technologies that are coming on to validate them, to cross, reference them and so on. So I think, something like this is easy to do. This is actually done with a simple camera, digital camera in the clinic. These days, of course, everybody has a camera. Everybody has a smartphone, everybody in the world pretty much, or a digital tablet. And so this can be done in the clinic. It can be standardized. It can be transferred to the home. Theoretically, we continue from home, these assessments. And towards that, I wanted to share one video poster that Lyle and Terry were also involved in. And I'll just share some clips of this just to show you. Now flash forward 2024, everybody's got iPhones and iPads, and this is all done with just remote smart devices. And simple exams like this is in the clinic, obviously, but simple get up and go task. There's so much information in this that Of course, many wearable sensors can pick up, whether it's on the ankle or the arm or the shoulder, but there's so much more rich data in these evaluations that you can see that the sensors probably couldn't pick up, that a neurologist might be able to pick up. In fact, some of these assessments with the digital analytics are finding things that neurologists might even miss, or finding new ways to interpret the data as to what really matters. So they're really becoming a neurologist assistant. And anyway, these are just examples. I just wanted to highlight that there is some value in simplicity and as much of a bridge as we can build to sort of a gold truth of a neurological examination is worth keeping in mind. Recently, actually, there's been a number of drug approvals that have actually been supported by video endpoints. And the use of video is kind of to do neurological assessments was sort of forced upon us by the pandemic. So there's a lot more comfort level now with tele-assessments in this area. So I think this is an area to keep an eye on. Obviously, you guys have a lot of magic sensors and stuff that you're looking at that are incredible. But I think in the ecosystem, video analysis, computer vision, AI analytics on this sort of data can be very powerful and very enabling. And I think, you know, what's going forward, it's part of it's about the devices and so on, but also how do we, you know, assess them? Can we actually develop a common neurological exam that serves across all indications that just standardizes the number of wearables rather than specific to the ALSFRS or UPDRS? You know, can we integrate using bridges like video across platforms? And I think we have to prove that they actually helped. So there should be some assessments later about did it really make an impact? Have we actually reduced costs and enhanced, you know, patient care? Anyway, those are my thoughts. I look forward to the discussion a little bit later. Thanks.
Thank you, Ajay. And now Nadine, are you? Yep.
Thank you, Lyle.
Yeah.
Let me just get my slides up. There we go. And you can tell me if you can actually see that.
Not yet.
Not yet. Okay. All right. Always a problem when I'm doing it on different computers.
That's what we need AI for.
It's a good example, right? You know.
Yes, and I'm, you know, definitely in need of some help here because I'm using a new computer for me.
Maybe while Nadine is trying to get her slides up, we can. So a bunch of people are sending me questions, and then there's some questions in the chat here too.
And you can email it to me so I can bring it up too, just in case.
Yeah.
But you know, so James, actually, we're not supposed to be asking questions, James. That's not allowed. But every time we see these talks, we all get excited, right? And he says, "This, Patrick, this is incredible," and is asking about, you know, the general population's lack of medical motivation. Will that play into the wearables? And you know, are there opportunities to build these into other devices for you know, for fun? And I kind of want to build on that. Maybe this one question before we go into Patrick. You know, when you talk about the profitability in this space, it is generally profitability for fun or for you know, athletics. It's not, as I understand it so far, profitability related to, say, pharma or drug development. And so, you know, do you have a comment on what that's maybe going to take? Because it's a different audience, right? It's the audience of convincing, you know, number one, companies that this is a good investment that's going to help their trials, and then number two, the FDA to pay attention to those.
Yeah, two very good questions there. I think the answer to both is that I think there's a lot of possibilities here. So, you know, my Center for Wearable Sensors, we have a couple of the funders are pharma companies, actually, because they see, you know, the power that wearables could offer to help improve the ability of their clinical trials to make interesting, you know, outcomes more easily accessible. So I think that's coming. The problem is that this is still a very much, and certainly in the medical sense, a very nascent field, and so there's a lot of things that we need to figure out, and there's a lot of improvements that we need to make on the sensors to make them more clinically accurate. The first question that was asked by James is actually, I had a slide on this, but I had to cut it because, you know, we had a short amount of time here. But integrating wearable sensors into objects we already use on a daily basis is the right way to go, especially for general population applications or for applications where people have, you know, low uptake for technology, right? And so my vision is I want to make some of these sensors so small and so unobtrusive that the user is virtually unaware that they're wearing their wearable. And I call this unawearables. All right, so this is when we integrate some of these technologies into objects we already use on a daily basis so that we get this rich feature sets without having to alter user behavior. And so I think that's a very clear path forward for this technology.
That's perfect. It looks like Nadine's got her slides ready. I think we can sort of return back to that too in the setting too of, you know, in therapy development, maybe, you know, passive data collection versus active and the kind of things where people are just wearing their clothing. What kind of data can we get for that that's useful as a, you know,
exactly
digital outcome measures? Nadine, are you,
Okay,
you can see it,
finally.
but you're not in presenter mode. Click maybe
Okay.
the,
Is that good?
that's the wrong monitor. Now we're seeing like your preview screen, switch screens.
Okay, that's what my presenter mode is
Oh,
doing.
Go back to where you were.
Okay,
Go back to where you were. I'm gonna, like, hit that again.
yep.
there.
And then there's a button there that should say switch screens or swap screens or something like that. In the
Uh,
upper.
Go back, Nadine. I'll guard
okay.
you.
Remember when it used to be that all that could go wrong was the light bulb going out in
Well,
the
now
overhead
everything
projector?
And you can see at the bottom, you know, where there are,
Okay.
okay,
Oh,
the next one. Yeah, yeah.
thank
There
you.
you go.
Okay.
Perfect. Okay. now
More coffee would be really important right now. All right, so I'm just going to put this slide up first because I am 1 month into being at CPATH. So I don't have my own slide deck, but I am going to tell you a little bit about what CPATH does, which is the consortium for Alzheimer's disease. And what you can see is, you know, really what we do is create tools and methods to increase efficiency of product development. And the reason we do that is that's information that the FDA likes to use in terms of how they develop guidance, how they develop letters of support, how they help work with researchers when you want to include all these different measures into your clinical trials and what's the best way to achieve these things. So CPATH has got a lot of experience in terms of moving forward with the FDA as well as the European Medicines Agency to create these types of tools and get regulatory approvals moving and regulatory guidance. Okay, so let me just get my button there. Okay, so I'm going to borrow a slide deck from my colleague Diane Stevenson. She's the exec director of CPP. And the reason I did that is because this is a good example of how we developed guidance and moved things forward with the FDA for a smartwatch for Parkinson's disease. So I know I've heard some chat in the early emails that, well, FDA isn't really interested. How do we talk to them? They have no, you know, view on digital health technologies. They actually have stepped into that area and they've been working there for a few years. They also have a digital health technology center of excellence at the FDA. So there's a lot of information on that particular website about what they're interested in, how they can help you as an investigator, bringing your information to them so that we can use these things in trials. So, you know, there's some fundamental groundwork already laid at the FDA. So for Parkinson's disease, getting evidence generation together for this novel Parkinson's tool, the Watch PD platform, the first and most important thing was engaging the regulators early, prior to study start if you can, because you can get a lot of really helpful questions from FDA in terms of how you think about you incorporate your device into the study design. And then it's, you know, a somewhat longer road where you get to study execution and then data sharing and eventually the clinical validation of your tool so that you can use your smartwatch or smartphone or you know, type of sensor that Patrick was describing, and it has a regulatory approval. So, you know, this is everything that was done in terms of going from 2018 to 2024 to get that draft guidance. So something that's of interest to all of you is the Digital Medicine Society, where they have frameworks about how you incorporate your digital health technology and presentation and have those discussions moving forward. So doing Watch PD, again, like many of the things that you've been talking about today, we're looking at multiple things going on. So what was clearly important to get this moving forward was to have it make sense to the participants, how they're going to use it as someone living with Parkinson's disease. So again, multiple functions that we were looking at: walking, balancing, tremor, spatial memory, everything you can think of that was verbal, auditory, movement driven, and also some executive function going on. So making this clinically meaningful meant that you really needed to listen to patients. And what made this go forward, I think, in a more efficient way was mapping patient-reported symptoms and getting a layout of that, of what was the most troublesome symptom, what was less so, and then something that was not an issue at all. And then, you know, work with that, figure out what you needed on the PD watch tasks, map those tasks back to the patient-reported symptoms. And then again, you do the activity that you wanted to measure, and then map all those symptom concepts back to the patient-reported symptoms so that they could articulate, I had trouble walking, I had trouble with my balance, my hand was shaking. So you have to make it relevant to the user. And I think Indu appreciates that, being in the IT space, that the client user is the person that you want to take care of at the top level. So this is some of the sub-study data that came out in terms of what are the health experiences that these people are experiencing in Parkinson's. So there's motor symptoms, which all of you movement disorder neurologists know backwards and forwards, and then the non-motor symptoms as well. All of these things can affect a patient, and you can see how important some of them are, how often they're repeated and recorded in these studies. Sharing the data with participants when you do one of these developments for a new tool is super important, because I think as researchers, we tend to forget that the patient is actually the living expert with the disease. They know things that, you know, we don't experience, and giving them the chance to understand how to best express that and communicate that can make a better product in the end. So the Watch PD participants, you know, getting that data, sharing it, made them comfortable. They wanted to know what was going on. They really wanted to see their data. And it helped refine the Watch PD process going forward. So, you know, just a couple of quotes from the participants with mapping. And I think given how energized and engaged the ALS community is, this would be a huge benefit going forward with any of the tools that you want to develop. So looking at, you know, the DHT landscape, Watch PD, you know, sets the stage for looking at new findings and how we can develop these things moving forward. And really, worldwide collaborations, EverythingALS has been all over that right from the beginning. Highly collaborative group reaching out across different expertise domains. And I think, you know, some of the things that you can look at are you know, doing crowdsourcing, working with other groups. A group that you should know is the Digital Medicine Society. They have a lot of great information in terms of standards, what you should think about, and I think how to communicate what you're doing as well. So you can, the Watch PD is something that is continuously being leveraged to get improved and novel endpoints incorporated into this particular tool. So, you know, if you look at this, like, because I'm a neurobiologist, I tend to look at things a little bit simply. So, you know, we look at Parkinson's disease and we see this continuation or continuum of the clinical phenotype of how things are changing that are, you know, they don't seem movement disorder related in the beginning, upset in terms of how the gut functions. And then we're looking at some changes in, you know, psychological effects, depression. We're also looking at, you know, as we go through those areas, we start to move into more of early movement changes. So there's some rigidity, there's some bradykinesia, and then these things start to get more and more and more advanced and more difficult in terms of having a person be able to live independently, move independently, have, you know, a solid daily quality of life. So when we looked at biomarkers of Parkinson's disease, you know, we have whole genome sequencing. We know gene mutations. We know some risk factors. We know that alpha-synuclein aggregates just from all the neuropathology data we have, and a synuclein aggregation assay, an in vitro test to see, you know, how much synuclein could be a problem already, depositing within the brain. And then we have dopamine transport imaging. So it's fairly basic, but as we look at the biology of these diseases, we know that immune function is a key contributor and maybe an early kick to moving into a neurodegenerative state. We know mitochondrial function can change. Lysosomal markers are definitely key in exploratory biomarkers that are being examined in many ongoing clinical trials in the neurology space right now. And again, as we move forward, synaptic density, protein aggregation markers as well. So those will continue to evolve, but it's like, how do we pick what's important? And that the FDA can actually wrap its thinking around and approve. So one of the quickest approvals was FDA writing a letter of support for the use of this alpha-synuclein-based biomarker in clinical trials. So this was that aggregation assay. This was, the study was really first published and developed around 2023. The FDA recognized the potential of this based on a lot of data that was submitted in its use in summer of 2024. So it was literally a 3-month turnaround for a letter of support, and that is highly unusual. Most of these letters of support can take several months. Sometimes they take a few years. So it's critical to realize that there's a pretty broad time frame where some of these things eventually result in an FDA approval or guidance documents. And I think what is important here is FDA likes data. They like lots of data. To get this letter of support, it was data from about 5 different clinical trials and 2 observational studies. So again, a lot of data that they can look at because they need to be sure that this is really rigorous, reproducible. So when you look at digital technologies of the future, and this was a slide that was put together by some of our sponsors in the consortium for Parkinson's disease from Novartis, these were some of the key things that they were looking at in terms of biological staging for Parkinson's disease going forward. And you can think, of some of the technologies you're talking about today, what some of these patches could also be measuring, other sensors that would be relevant to ALS. So, current state of the art, if we look at gait analysis, when we were emailing yesterday, I was asking, what's the question we're going to ask? What's relevant? Because FDA will look at these devices, but I think if you want to use something in a clinical trial, they want to know what it is you're measuring, what's the relevance to the disease, and can this be measured in a way that it can be incorporated in a clinical trial so that it may be a diagnostic marker, it may be a prognostic marker, it may be an endpoint, but something that has really robust measurements and context of use. So when I think simplicity, I'm thinking for ALS dorsiflexion, because that is something that happens early and is changing and is progressive, and you eventually get a slap walking type of gait. So this study is looking at the state of the art in terms of what they have an acceleration sensor and a neuromorphic computer, which I will not attempt to describe. But what is key is, what are you going to ask that can be used in a clinical trial? And I think, we shouldn't forget that FDA does like to discuss these things. They do want interaction. Talk to them early. I think, in the drug development space, we have patient focused drug development meetings, and it's really a great listening session for the FDA. Patients are there, advocates, industry is there. They get a sense of what's going on, what's really key in the field. I think it might be interesting to see if we could get something similar that would be a patient-focused DHT type of meeting. And I don't know if they have this space yet, but I think it's worth getting those conversations going, and maybe we could move something forward that way so they could listen to your community, your needs, and where you are right now. But don't think that the FDA isn't interested. They are, but they need to learn how to think about these technologies the same as we do. And you need to calm down on, what do I want to use it for? Context of use is always going to be critical for whatever technology you're developing. And that's it. welcome any questions.
So thank you. That was really wonderful. And you know, we're already running late, but we have to do some questions because there's some questions that have come in, and I want to highlight some of the important stuff you guys said. So Indu, Jen, you guys can just like cut my pay in half, take it out of my, whatever you're supposed to, you know, the half of the privilege of working with you guys, I guess. But you know, I think that sort of building off of what Nadine was just talking about, so you know, people living with ALS, people with lived experience of disease are critical to this stuff, right? And we had a question from Doug Hill in the chat that says, you know, our upcoming trial designs considering these wearables for biomarkers, and Indu replied, yes, of course, but here's the rub, right? So what and how do we pay for it? And over the past couple of days, we've heard a bunch of amazing talks from small pharma about really fantastic drug development in this area, right? But small pharma and even big pharma will tell you when they're planning for a trial, if the budget has a crunch, the first thing to get cut is biomarkers, even if that's just collecting biofluids. And so, you know, I think a really important question for us as a community is, how can we potentially, you know, EverythingALS or some of these clinical trial networks like the Healy, you know, how can we create an infrastructure where we can incorporate these things and it isn't a huge financial burden to all these separate companies, and you know, and at the same time that gives us a way to test them across platforms. And I think that's really important. And I have a specific question, I guess, for Ajay about this, which is, you know, you work with a lot of small companies, right? If a company comes to you and you're thinking about working with them, how do you decide what's good? And what could possibly EverythingALS do to provide some infrastructure to help you figure that out?
Yeah, I mean, there's such a proliferation of these technologies that it's often hard to tell some companies apart. You know, they have a different way of measuring something, but so I mean, just in my career, I'm focused on what can't be measured and who's going to crack that. You know, there's so many ways to measure Parkinson's. I think we still have remotely, at least, a hard time measuring rigidity, which is one of the earliest, you know, signs. So, you know, I look for people that are actually trying to solve problems and create solutions for where we don't have something, rather than a me too, you know, that's a little bit better. I don't know if that's helpful, but that's just a general, because that's what's going to solve a problem. That's what people are going to pay for. That's what's going to make a difference.
So, I mean, it gets back to sort of clinical meaningfulness, which, you know, Nadine just touched on a lot, and it really is the key, right? And the problem that we talk about a lot in this space is if you're creating some new digital tech that measures something, how do you prove that what it's measuring is actually clinically meaningful? And the analogy that Ernest Franco came up with that I love is, you know, the person who invented the first thermometer, like, how did you prove that it was measuring something meaningful when all you had to compare it to was like holding your hand to your forehead and saying, do I have a fever, right? And that's where we are in a lot of this space. And it's, you know, James mentioned that in the context of listener effort. How hard is it to understand what somebody's saying to you? Seems like something that should be meaningful. But I think we still have a long way to go into figuring that out. And Nadine, I like your idea of perhaps having a, you know, people with lived experience based conference. I'll tell you, one of the other hats I wear in this space, right, is that I chair the programmatic panel for the Department of Defense ALS research program, which funds a lot of these studies. Not just that, you know, we have ways to fund digital biomarker studies and really anything related to clinical therapy development in ALS. And we require that there's people with lived experience on these grants. But just providing grants isn't going to answer this. I think we need an infrastructure that lets us do it more broadly and have, as we said, sort of a fast fail system. And so I guess with that in mind, I think it's important that we think about, like, what would be the components? You know, do we need standard data sets? Do we need voice data or wearable data that can be used to test out, you know, different algorithms, and really figure out what we should be incorporating into these trials and how do we measure their effect? So,
you know, I'd suggest, you know, look at all the things you test as a neurologist when you're diagnosing something with ALS, and, you know, you're looking at things that you are usually going to see progression in because you see the early signs, and then it progresses, and that gives you confidence that, yes, this is ALS. It's not multiple system atrophy. It's not something else. And those are clinical signs, phenotypes that the FDA recognizes. It's in the clinical literature. There's lots of evidence for that. So they get a little bit more comfort around that when you build a sensor or device that reflects what does that look like in, you know, digital data.
And Nadine, what do you envision? How could CPATH help us? I mean, I know it's an evolving sort of an idea, but what role could CPATH play in this for us?
So what we start to do is initiate some of those conversations with the FDA, start to get them more aware, a better understanding of what the landscape looks like, what the data is, get questions from them in terms of what else do they need to see, what more can you provide. You know, for the Parkinson's watch, really huge benefit was all the clinical trial data that was collected. And, you know, there's a lot more people with Parkinson's than ALS. So again, you're looking at this from a rare disease space and the amount of data that you can collect. But, you know, sometimes you can get some simple readouts for yourself to give you an idea of where the data would be going that you can illustrate to FDA. So like I've got a smartwatch that I use for running. It does a lot of cool things that would be, you know, similar and invaluable to ALS. It does gait, it does my pace, changes in stride length, vertical oscillation, heart rate, for a pretty low price, like much less than some of the very complex ones you do. So, you know, look at something that it's very basic so they can figure it out. But CPATH has a lot of these conversations. We have steering committees within the FDA that we work with because we have several consortia in the organization. So it's not just Alzheimer's and Parkinson's, there's diabetes and others. And I think, you know, it'd be really valuable for us to move forward in something that is a digital health technology conversation and lay the groundwork for them to have more information, a better understanding of what you're trying to do so that they can figure out, okay, how do we write guidance around this? How should it be done moving forward? Because it's new for them too, but they really want to learn so that they can do effective guidance.
And one last question, and it's I guess sort of for all three of you, and it gets to this idea. So the data, if you're recording my face on a phone app, or you know, or recording my voice, or something about me on a wearable, you know, it seems that there are some patient confidentiality, you know, protection sort of concerns that are different than maybe just drawing blood and having de-identified biosamples, right? I mean, it's somebody's face, it's somebody's voice. Ajay, you know, I like you, I'm very excited about the cell phone based video capture, mostly because I think of it as a clinical trial equity issue, right? It allows people who have a cell phone wherever you may live, and that's everybody, to participate in these trials. And it doesn't require expensive wearables or, you know, living near a brick and mortar center. But what have you guys experienced, whether it's in an academic, you know, research setting or you know, working with developing these techs or with FDA, you know, how can we deal with this idea that we're recording somebody's video or sound on their phone?
So, you know, we're actually directly dealing with this right now in a project. There is consent people can give. There is ways to blur out the face. There's also the HIPAA compliant, you know, how you transfer the data. So there are some guardrails in place. Society is also changing a little bit if we opt in or not. I mean, frankly, there's not that much difference between a nanny cam and a granny cam. You know, you have to get permissions, and I'm actually surprised that a lot of the ADTs and home care, you know, home service companies aren't actually in the biomarker space, just providing streams of data. So you know, I think there's ways to handle it if people can opt in. There's some compliance issues. You can actually blur out identity. It shouldn't hold us back, I don't think, too much, but we do need legal people involved and the FDA involved in this conversation, and patients involved. If they want it, then why not?
Patrick, do you want to make any comments on that, or final comments on the other things that have been brought up?
Yeah, so whenever you're developing new technology for human interaction, you need to think of privacy, security, and things like this from the ground up on day one, right? And that's particularly true when we're talking about, you know, this unawareables concept where we want to put sensors into shirts and stuff like that. We need to have good ways to make sure that patients are offering their consent to use these things, which is kind of the opposite of what we want unawareables to be. So this is a challenge, I think. But that's where we have to interact with our ethics colleagues and our colleagues who are working in design. You know, as engineers, we tend to not really care about, you know, aesthetics, but and usability. But this really matters in this context.
Thank you all. That was really wonderful, and thank you obviously to Indu and Jen. Oh my gosh, this has been an amazing few days. So thank you, thank you, thank you. And I think Indu, we're going to move on to the next panel. Is that?
Yes.
Okay.
Yes. So thank you very much. And let me bring up our next panel. As you can see here, it's from nice to have to must have, and what will it take? So joining us are, you know, different companies here, but moderating would be Carl Sille, and Carl is a clinical and academic neurosurgeon with background in Parkinson's disease with gene therapy and neuromodulation clinical trial surgery, with early interest in IP generation, translation, and embedded objective measures. When I ran into Carl, he's done so much work in digital biomarkers for over 10 years now. So I was very thrilled to actually, and honored to have him join the ALS journey here and to participate in scaling these technologies and to broaden our clinical application to positively impact, you know, clinical care and the digital biomarkers. So with that, I will invite Carl to moderate the session.
Well, thank you so much, Indu. Are you all able to hear me okay? Great. It's my real honor to be part of a fast-moving group that really wants to make a difference and take that right to patients, because I think a lot of us have had the experience that's been discussed and the opportunity to scale it. So in this session, we're going to talk about ensuring that AI delivers value, going from nice to have to must to have, and what it takes to deliver value in the treatment and identification of a cure for ALS and betterment for the patients. So in this panel, we will touch on AI-driven solutions, the challenges to scaling the technologies has been touched on already, ensuring clinical precision, maintaining it, usability and adaptability, and overcoming the regulatory hurdles. And then some highlights that we could take from other areas, cross-learning from successes in other areas. So one of the things that we're thinking about is that AI really should be a source of good. And we're hoping to hear from each of the presenters today how their company or their institution sees this AI as a force of good and benefit and how it's implemented. So it is my privilege to introduce our distinguished panelists, including Dr. Ernest Frankel, professor at the Massachusetts Institute of Technology, renowned researcher in systems biology, with sophisticated work in computational and experimental approaches to unraveling complex biological networks with the goal of advancing precision medicine. By employing artificial intelligence to large-scale biological data, his team provides deeper insights into disease mechanisms. And in the realm of ALS, his contributions have been pivotal in identifying potential therapeutic targets and biomarkers, paving a way for personalized medical interventions in a way that could really advance and alter the disease. We're also joined by Dr. Alvaro Pascual-Leon, who is a professor of neurology at the Harvard Medical School and medical director of the Deanna and Sydney Walk Center for Memory Health at the Hebrew Senior Life. He has had a pioneering work in brain plasticity, non-invasive brain stimulation, and exploring neuroplasticity and promising avenues for treatments that potentially could find ways to monitor and slow the disease. We're also joined by Dimitri Starson, who is a leading expert in computer vision and applications in healthcare. There have been many discussions about nutrition, diet, and chemicals associated with neurodegenerative diseases, and his team has expertise in the development of tools that can monitor disease through visual access and even looking at visual biomarkers, including food and nutrition biomarkers. And Dr. David Sunderman-Uft, DSO, is the founder and CEO of Modality AI, a digital health company specializing in AI for data-driven platforms that communicate, interact, and can potentially track motor function and ALS and monitor things such as speech and other motor areas. So please join me in welcoming these distinguished panelists, and we will have probably abridge it a little bit if everyone could work, but we're supposed to have 10 to 25 minutes for the presentations and the remaining time, 15 to 25 minutes for Q&A. So we would invite Dr. Frankel to begin the session.
Hi, thank you very much for the very kind introduction. Let me see if I can get my slides to share. Let me know if you're seeing that. Someone? Yeah,
Yes.
a thumbs up. Excellent. So I'll try to keep this brief. I did want to give sort of as an academic an overview to some of the real issues and the concepts that we should be thinking about as we go through these talks, of course, mixing in a few results. So, where AI can and can't help. And I think that the first thing that I think we're all in society getting to be more and more aware of is how data hungry AI methods are. And this is kind of broken out into the popular press from the academic literature with these lawsuits, because to train something like ChatGPT, you need more data than's even available on the internet. And so there are all these copyright infringement cases because to train the models, the companies had to sort of dig into things that may be on the fringes of legality. So as we think about this in the context of ALS, where are the big data sets and where can AI make an impact? The next slide is some audio. If you don't hear anything, someone should shout and tell me, but hopefully it'll play.
Don't hear anything.
All right. Well, what that slide was an automated voice of mine that had been produced by totally free tools that you could access on the internet. And the point here is I used a large language model to write the text, and then I fed it into a large language model that produced automated voice. And it sounds not bad, quite honestly. It's a little bit flat, but it's really very good. And why can we make these synthetic voices so well? Because there's just so much audio data out there, right? And so that goes back to the key point. Where there's a lot of data, you can have a big impact. So where do we expect to have enough data, and what kinds of goals are we setting for ourselves? And so I think there are two goals that we should be thinking about. One is in biomarkers to monitor progression, but the other one is in subtyping the disease. And here, I think that the thing that we don't often talk about, but I think we all recognize at some level, is that ALS really isn't one disease any more than cancer is one disease. It's a clinical diagnosis that shares a lot of common symptoms and maybe some common mechanisms. And so if you look at the genetics, we all know that for the 10 or 15% of people with ALS, you can find genes that cause the disease, but there are a lot of different genes and they don't seem to fall into one common pathway. And the drug that's going to work, this tofersen, works for the SOD1 people, but will probably not work for any of the others. In terms of clinical presentation, it's been known for a century that it's clinically diverse. And then in terms of progression rate, you've got a decade or two of spread. So we need to think about how we can use AI to find subtypes. And that's something AI is really good at. It's the same way that AI can help figure out, what kind of movie you want to watch next or what you want to buy next to send you the right ads. So AI is very good at subtyping people if we can feed it enough data. So I've been privileged to work with a really great group of scientists in a program called Answer ALS that generated a lot of data from people living with ALS and controls. And there are about 1,000 people with ALS who participate in this program. And we collected their clinical data, took biofluids, and then from those from the blood, we're able to make induced pluripotent stem cells and generate a lot of neurons from that, and then put those through various kinds of omics. And that provides a rich place for data. Now, the size of the data set, it's still 1,000 people, which is nothing like the size of the internet of voice data or written data. So we're still in a regime, we're data limited, but we're getting in the right direction. And all this data, all these data are available if anybody has interest in this stuff from the Answer ALS. I think they're calling it Neuromine these days. So what we've been interested in is different ways that you can subtype ALS. You can go directly from the blood to blood biomarkers. You can make the blood into iPS cells and then to motor neurons and then do various kinds of omics. You can take those cells and do tests on them in the laboratory. And then, of course, we've got the clinical subtypes. So maybe I'll start with the clinical subtypes. Early on when we were studying ALS, we thought, oh, okay, we'll take the ALSFRS rating score, we'll plot it for patients, and we'll separate our data from Answer ALS to fast progressing and slow progressing people, and it'll be pretty straightforward, and then we'll go do interesting stuff. And we discovered that that's actually a really hard problem because you don't see people for very long when they have ALS, so you've got a short period of data, and then the ALS rating score is based on questions that the answer may vary from day to day, so there's a lot of noise in the data. And when you're done, it's pretty hard to tell which of these people was actually the fast progressor and the slow progressor. So AI was very useful here. This is all published. We were able to then take I call this a spaghettigram. It looks like you threw spaghetti on the floor, take the spaghettigram of ALSFRS-R trajectories for people in the Answer ALS study, and use AI to group them into patterns. And these patterns had some interesting properties. You can see right away that most of them are highly nonlinear. And that's true in clinical trials as well. So these are data from the ceftriaxone trial that were available from the NIH. And you can see how highly nonlinear some of these patterns are. And obviously, it's going to have big implications for how you interpret results in clinical trials if you model things with a linear trajectory, but in reality, it's nonlinear. So that's AI directly applied to ALSFRS-R. The question is, can you do better than the ALSFRS-R? Because it's got a small dynamic range. So here we're showing the questions that are used for speech, right? It's a 0 to 4 scale. And it's not very granular. So there's no 3.4, 3.5, 3.6. And the scoring, as I've said, is subjective. So today you might be a 3, and then tomorrow maybe you're a 2. So I've been very fortunate to work with EverythingALS in this. Lyle, James, Kelly Norell from IBM, and a great team of people working directly for EverythingALS. And so we've been able to collect a very large data set. Again, size of data set is really the critical thing, gathered over more than a year, more than 1,000 hours of data, and then try to make some sense of this and see whether we can solve this problem. Lyle already used my analogy, so can we do better than the gold standard, right? We want to make the new thermometer, but we have to compare ourselves to sticking your hand on someone's forehead. So this paper is available on the archive because it's going to go through pretty quickly. But we said, okay, well, what can we do? Like, we need to do something, right? This is an urgent problem. And the idea emerged through talks with Lyle and James and others that, well, speech-language pathologists who work in the clinics with people with ALS actually have a rating scale that's better than the ALSFRS-R for this. It's listener effort. How hard is it for someone to understand a person with ALS when they're trying to speak? What's the point of speaking if not to be understood, right? And so we could use the listener effort. And what this paper shows is that when you have speech-language pathologists independently evaluate how hard it is to understand somebody, you get very, very consistent scores across the speakers. And over time, if you randomly resample the data and feed it to the listener at different intervals, they tend to score it with high consistency. And then we're in a position where we can use AI and see, well, can you take the person out of the loop and just use AI to predict it? And we get really, really nice results where we can show that we can actually use a totally automated process to take that speech and use it to quantify how hard it would be for a human being to understand them. So we're very excited about that as a potential way to improve on the ALSFRS-R. I'll zoom through these final things because I think they're a little bit less relevant to today's talk, but we've been able to use the multi-omics generated Answer ALS to find clinically meaningful signals in data from cells that were derived from cells, that were derived from blood, that was taken from the patients years before. And I think this is really super exciting to show that there's molecular signal that persists, and that's available here at this QR code. So that's already published. And then we have unpublished data where we take the blood directly and do untargeted metabolomics, and we can find subtypes that correspond to things that are present in the multi-omics from Answer ALS and also in the clinical data, and hopefully that'll be coming out in the not too distant future. So the last piece where we think ALI can make a difference is you take those cells that we've derived from the patients and put them into experimental conditions where maybe you can tell a difference between a person with subtype 1 and subtype 2 of the disease, and then use AI to that, and then come back and see if that relates to the subtypes that we're identifying in the clinical data or directly from the blood. So that was the quick overview. I hope that was fast enough. Just to summarize then, we think that AI has a big role, but it's going to be always limited by how much data we have. We're in a fortunate position when we can do remote data collection like EverythingALS did. So that really opens up new possibilities for AI. And then, of course, from the Answer ALS study where we've got all these cell lines in the lab. And I'll turn it back to our moderator.
Thank you very much for that really wonderful overview and really diligent and pioneering work integrating so much. And we look forward to the question and answer section. I'd like to invite now Alvaro to speak, and we look forward to hearing your presentation.
Thank you so much, Carl, and thank you to EverythingALS for the opportunity. Let me see if sharing the slides works. So as you heard, I'm a professor of, can you see the slides okay, by the way? Yeah, thank you. So as you heard, I'm a professor of neurology at Harvard Medical School, but I'm here also in representation of Linus Health and want to tell you a little bit about the approach we're taking there to try to address the question of how can we leverage technology, neurotechnology in particular, to enable clinicians to do what we do better, to enable and empower patients to take control of their own situation and to track things end to end from detection to wraparound care. The approach we have taken at Linus Health is to leverage digital diagnostics rather than reinventing wheels, capturing different signals with great precision, emulating a little bit what we might do as clinicians, have a sense of the picture of the pattern that the patient shows us and do sort of pattern recognition to classify, but do so with more precision, hopefully, than what clinicians can do by leveraging the precision of the technology in capturing the data and the AI analysis of those signals to extract predictions for different aspects. And we try to do that with very easy, straightforward, short assessments in such a way that it might be particularly easy to integrate it in workflow. The idea behind it is not a new one, obviously, and it's something that you all share. We have been hearing about throughout the sessions. In neurological exam, in cognitive assessment with paper and pencil, we look at the output of what a patient is asked to do, and we know that there is limited sensitivity, but we also know that when an expert clinician does such an assessment, there is a lot of information that is extracted that Edith Kaplan in cognitive neuroscience, for example, conceptualized as pointing out that the key information that the expert clinician observer extracts is metrics of the process, rather than looking simply at the final result, is how the patient does what they do, that we capture signs in a more sensitive way. Of course, that's difficult to do because it takes a lot of years of training and practice and experience. It also requires the expert doing it. It's not something that can be easily scaled to having a medical assistant do the job. However, technology should enable that type of process metric capture with great precision in the hands of a relatively untrained person. Essentially, I like to think of it as putting the expert in a box, as it were. And that requires a lot of issues that Ernest has already pointed to in terms of validation of the process, of the analysis, and so on. But it provides at least a means for capitalizing on the power of technology in a way that I would argue to this day we haven't fully done. So what Linus is trying to do is capture not just one signal, but multiple different modalities of signals in a integrated platform to be able to, by integrating the signals from drawing and motor control of fingers, gait and balance, speech and language, or eye movements, to integrate a multimodal type of assessment. To highlight one of them, speech obviously illustrates the example of the approach rather nicely, and it's what a lot of the work in ALS has focused on so far through oral analytics. There are motor signs and motor components to the speech. There are respiratory components. There are cognitive and behavioral components. It's easy to capture, or relatively easy to capture in a mobile kind of way. And the richness of that signal can be then analyzed in such a way that it gives reliable, repeatable, scalable metrics in a manner that is faster and arguably more precise than what the even expert ear might hear. The idea, again, in order to make it not just an assessment, but actually something that can go from the academic development of a tool to the implementation in clinical trials and in clinical workflows, is to offer a platform that captures the signal, that provides the analysis and the metric of computation, and that returns the results in a way that can be repeatedly replayed by the clinician, can be modified, can be double-checked, and can be translated in actionable recommendations for the patients. So to give you a sense of the sort of overview and to get into the different components where we apply AI to get us back to the sort of topic, one can do remote evaluation on the phone or on the web, use AI to extract different engines of analysis for those multiple metrics captured and assess different aspects. The same can be applied in clinic screening in a way that can be scalable easily because of the need for minimal additional technology that people may not have, and because of the fact that it's not dependent on expert training. The clinicians can then replay it and reassess it. AI can also be used, and it's also being used by us, to personalize the recommendations in a way that translating the findings into actionable recommendations to the patient, their caregiver, or the clinicians, as well as to guide in a workflow manner the next steps, aiming to guide decisions regarding therapies or chronic care. In ALS specifically, just to wrap up and at least finish with a little bit of data, looking just at the speech vitals, which we've done, getting a breakthrough designation in ALS from the FDA, the findings, I agree with Ernest. I think at the end of the day, we need to make sure that we measure against the clinical standard and show not only that we can match it, but ideally outperform it. And in this case, we have evidence that we can outperform the ALS FRS-R, given the granularity and the greater sensitivity of the captured signals. And importantly, we can link it then to recommendations that are appealing to patients, that translate into better quality of life of patients, and that in a workflow that can be easily integrated in the clinic, not only in studies. Maybe I'll stop there just to give you an overview, and then we can expand on things in the questions that may come.
Thank you so much for that amazing work. It's really, really wonderful to hear that. We look forward now to hearing from Dimitri Starson and our next presentation, and we'll have time at the end for questions.
Wonderful. Thank you so much for inviting me. And for the opportunity to support everything ALS and the ALS community. It's been just tremendous pleasure. Please let me know, are you able to see the presentation and to see my screen?
Yes.
Yes.
Wonderful. Yes, so let me share a little bit about myself and the company. So Passio AI, we are a spin-off from the Stanford Research Institute, SRI International in Menlo Park. We started the company by focusing our AI development on solving the challenge of nutrition tracking in fitness, healthcare, and several other industries. So as we all know, nutrition plays an incredibly important role in all diseases, in all healthcare, and especially in neurodegenerative diseases, ALS, Parkinson's, everything. But historically, all of the efforts around nutrition, they've kind of been random and not grounded in data, because data collection and understanding of what members are eating has been extremely difficult. It is still not easy, even with the AI tools, with the AI magic that I will share with you, but it is becoming easier and easier, and we are working every single day to address this. So we started the company with the plan, the vision to create AI-powered tools that make nutrition tracking and AI-powered nutrition coaching much easier. So let's make it easy to track, and let's give everyone an AI nutrition coach that will help them with optimizing their nutrition, getting answers about nutrition, and building truly personalized nutrition care regimens. So with the tools that we provide, my company, we provide components and fully functional modules that integrate into enterprise healthcare applications. Like EverythingALS, we've been very privileged to be working with Indu and Indu's team to integrate AI nutritionist capabilities into the EverythingALS app, and we will hopefully get it into the hands of the community in the coming weeks and months. But we power other healthcare applications, including our AI nutritionist is currently embedded in tools like Sydney Health by Elevance Health. We power applications from ShareCare and quite a bit of other applications in the health, both across healthcare and fitness and weight loss industries. So what these tools allow us to do is to improve member engagement, help members with understanding their nutrition, collect detailed nutrition reports, which are very important for all of those studies that other participants mentioned, right? So when you are collecting biomarkers, when you are collecting voice logs and analyze the evolution of patients' journey through their life, it's very helpful to collect data points on what patients are eating, and that's essentially what we help to do for the community. Collection of nutrition logs that can also be very important, as we know, in clinical trials, and understanding the interaction between what people are eating and what medications they are taking. And obviously, the next step after we do this understanding is to go and start creating treatment plans and care improvement plans. So from the to validate the importance of this, one of the quick short studies we did, we asked a few hundred healthcare professionals whether getting access to patients and member nutrition data could help them with creating better standards of care, better treatment plans. And the vast majority, over 90%, answered yes, but less than 14% of the population that we asked said that they had any visibility. So we know that there's this just massive gap between what people are asking for and is it just me? There's a little bit of
If everyone could please check their microphones are muted, please.
Thank you. All right, let me move on. So there is this massive gap that the tools that we adventured to create are filling, and so with the AI, there are several things that we are able to do right now that's not been possible really historically. First, from the nutrition tracking perspective, the entire food tracking industry initially was rooted in manual diaries and very, very kind of surface level surveys. Now, with AI, a person can enter their meals, in some cases almost automatically. In some cases, you can just put a camera in the kitchen and it tracks everything you eat. But on a less intrusive way of doing this is to provide people with a nutrition tracking app, which allows several modes of data entry, from computer vision to voice logging to manual search. That essentially covers all of those modalities. And I will show a set of quick demonstrations where we developed this technology. You can log your meals right now with just a photo, or with a real-time video. You just point your phone at the food and it scans all of the foods in front of you. Or you or your caregiver can talk into the phone and say what you had in a day, and it logs, creates the full log just from the voice. So we've been trying to perfect this technology for many, many years. And as you can, as you know, there's a lot of complexity in this. Like, this is a very, very simple example. We snap a picture and it basically tells us a recipe. But imagine what happens if there are uncertainty about what's inside of the food. So here, for example, if we take a photo of a burrito, AI needs to be smart enough to ask what kind of burrito this is. And so here it clarifies, is it a pork burrito or is this a chicken burrito? So basically thinking through those degrees of complexities, like another example would be taking a photo of a cup of coffee. It recognizes, it knows that this is coffee, but for real nutrition log, the important part here is to ask, did you add sugar into the coffee? So at Passio, with our effort, we've thought through and went through all of those edge cases. Many people want to scan their logs on Sunday, create the entire meal plan. Some people log one by one. Some people log the entire plate with food. And that's essentially the tool that we created and integrated into the everything, and are actively integrating into EverythingALS app. Another mode of input is voice. About 50% of people are logging with computer vision. Another half is logging with voice, where you can narrate your entire meal or your entire day of food with amounts, with times of when you ate them. And it basically, AI right now analyzes and creates the food log and adds this log into the system. And what we combined in our tools, both giant databases and different ways of tracking. And after the when members actually are able to track their nutrition, the next step is for them to start modifying their nutrition. For example, for the ALS community, there is an assumption that Mediterranean diets or diets rich in omega-3 can be helpful. And so what we are starting to create with our registered dietitians and with the treatment community, create a variety of meal plans that patients are able to adhere to, and in combination with prescriptive meal plans, prescriptive journeys, and tracking of what patients are eating and tracking of other biomarkers, hopefully over time, we will finally be able to answer a question: what is an optimal diet for an individual? Not for a category of people, but for an individual, and optimize this on an individual basis. And the last component of why it is so important, again, to create those personalized plans and to create studies, to create the understanding, better understanding of the outcomes based on nutrition, it is important not just to collect data, but to properly present it and share it with the medical community, with the research community, so that if we are tracking blood biomarkers, if we're tracking voice biomarkers and then different outcomes, another very, very important piece of data is nutrition. And that's essentially what we are excited to bring into the world. And I believe that this is becoming not just a nice to have, but a must have, because as all of us know, nutrition plays a vital, major role in the care and in the outcomes and in what we can do to support the ALS community. So thank you. I would be excited to take questions during the Q&A session.
Thank you very much for that amazing and wonderful work. There's so much that can happen with that, and I look forward to the questions. Next, David, we'd like to hear from your presentation, please.
Absolutely. Thank you very much for the invitation. We've been partnering with EverythingALS since 2020, so it's a significant privilege for us to talk a little bit about what we've been doing with them. We focus as modality also on multimodal assessments. That's why we are called modality, and specifically on the support of clinical trials in ALS and other related diseases. As you know, many of the clinical trials, specifically drug trials, fail these days. That's something which, of course, is in the way of moving the development of these treatments forward. In fact, phase 3 trials, for example, 97% of ALS trials have failed. And we are focusing as a company on supporting the developers of drugs and treatments in their journey by providing assessments that can help them really make sense of what is going on with their patients and their interventions in those studies. And we've heard from James Berry, from many others over the course of the last 3 days about the shortcomings of today's assessment, specifically with the primary endpoint ALS-FRS-R, that is not very sensitive and not very responsive. Ernest Frankel was also alluding to this earlier today. So what we do, we are inspired by what actually happens at the clinic when neurologists and speech-language pathologists interview patients. And what we do is that we emulate this process by using a virtual guide, Tina, which many of the people in the audience may have already seen in the past. Many patients that have, for example, gone through the speech study with EverythingALS actually have used Tina in a repeated fashion. She interviews patients and does so in a way that is inspired and very similar to what a speech-language pathologist or a neurologist would do at the clinic. She asked people to respond to certain open-ended questions, describe pictures, repeat sentences, do a reading passage, diakinesis, and other things. She also asked patients to describe their problems in their own words. In fact, she, in doing so, measures how patients function and how they feel, both, which is very relevant, of course, for the regulatory concerns and for the concerns of the drug developers specifically. So we look at speech, language, facial movements, body movements, cognition, as well as I mentioned, open-ended questions about their symptoms. And doing so on your own device. What we do is we provide a web application to anyone that has a mobile device or laptop. Anything that's web connected is actually compatible with the modality and Tina. And you don't even have to install any software because we use web technologies. You don't have to remember passwords or credentials. We send a link to the patient and they click on the link. Next thing that happens, you can talk to Tina, making it fully accessible to pretty much everyone who has a device and can interact in that way. Just very briefly, I mentioned we are supporting clinical research exclusively. We have been used in many studies in ALS, particularly in Parkinson's disease. Interventional trials, randomized control trials, many observational studies in which we are one of the leading observational studies, of course, the EverythingALS speech study that we have been supporting over 100 publications in over 150 sites across the globe in 17 countries. And we have touched the lives of over 80,000 patients, almost 80,000 patients with TINA today. And that's in a nutshell. We presented yesterday a full-blown presentation, including the scientific data. But for today's sake, I wanted to keep it brief and look forward to the Q&A.
Thank you very much for each of the presenters. We have about 10 minutes, and I wonder if we could think about questions in areas like concept to deployment, clinical precision versus end user experience, scalability, standardization, and future directions, innovation, call to action. I wonder, Dimitri, what were some of the key challenges you encountered when moving your technology from concept to deployment?
Yeah, so nutrition tracking as an area of data collection historically has been insanely difficult because tracking food is not something that people normally do. This is not as easy as wearing a step tracker, right? It is a very substantial behavioral change for people. And unless people are intrinsically highly motivated to do so, it is historically it's been very difficult. Not everybody would pick up a journal and write things. So even as we started to introduce tools like snap a photo and it logs the meal for you, or narrate it with voice, we are seeing higher and higher adoption. But until this technology starts properly working, the adoption is extremely difficult because nutrition data collection or nutrition tracking in and of itself is very much counter to what people normally want to do. So this is not something that's ingrained in our nature. And so going against kind of what people normally want to do in their daily lives, it is a challenge. Now, it is significantly lower challenge when we bring the right tools, right? If you can narrate either yourself or if you're a caregiver, if you can just narrate in the evening everything that a member ate, reduces the barrier of entry. If you can snap a photo, reduces the barrier of entry. And then even after we reduce the barrier of entry, then there is a second challenge that comes into play. There's never been highly accurate nutrition sources of nutrition data. So for many years, we've been collecting large data sets of just food information. Even USDA that provides certain nutrition for thousands and thousands of foods, it's not 100% accurate. And so building and the nutrition industry, the food industry, adds new foods with a lot of new ingredients all the time. So this is a very, very dynamic and evolving area that needs to be properly, that requires just consistent high degree of effort. And the third component, the third part of this, bringing this into the community, because this data has never been collected and there's never been really robust enough studies that are not contradictory to each other, it's very difficult to provide AI-driven recommendation. It's possible, and we are definitely doing this. It makes already an impact, right? But for example, let's say we've got a patient and we recommend them to transition to Mediterranean diet, right? Should this Mediterranean diet include fish or not? Should it include, should it be 20% fat or 25% fat, right? There's the complexity of the space is so huge that providing robust recommendations is not an easy task. All of this becomes easier the more data we collect, right? The more studies we provide, and we're trying to aggregate this into the robust AI systems. But the scale, the entirety of the scale of this challenge, and combined with the human nature of not wanting to eat broccoli, not wanting to take photos of their broccoli, but rather wanting to eat Twinkies and never take photos of them or never record that food. Those are the types of challenges that we are fighting. And in addition to this, the last thing I would say that hopefully is changing right now, there is also the incentive structures are not yet perfectly aligned in our society, as we all in the healthcare industry know. there is just giant food healthcare industrial complex that does not make it easier, to say the least, with all of the complexity of foods and additives that are being put into the food system. So that's kind of the last piece. There is a little bit of misalignment of incentives, but that would be a much longer conversation.
Well, thank you for that. That's an excellent segue to maybe Alvaro, could you touch on how you balance the need for clinical precision with usability for consumer or end users? Because he was talking about what their burden was and whether they were tracking correctly. But I would like to hear your thoughts.
It's a great question, Carl, and I think it echoes many of the points we were just hearing about, and everybody has been touching on, right? So adoption and adherence with these metrics is complicated, and it's complicated just because clinicians are used to a certain habit. We like to do what we like to do. We may be willing to accept that it may not be the best, but it's what we're used to. And patients in turn have a hurdle in doing something completely new. There is an appeal initially and then a hard time adhering to it. So I think that the emphasis that you're highlighting of making it as friction-free as possible, as easy to incorporate in either clinician workflow or daily life becomes really critical. And of course, there are low-hanging fruits there, short assessments, easy to capture, forms of information that are easy to share. All that can be very valuable, and I think that we need to use all of them. But I think there is another aspect that is worth considering, which is that as we think about the importance of having reliable, valid metrics, we generally think that the way to do it is through a study. And of course, there is great value in that, and it's critical. But there is also an opportunity that I think we often underestimate of using approaches in clinic, in real world, and to benefit patients now with the limitations and the appropriate disclosures, while leveraging that information to learn fundamental aspects and gain novel insights. And I think that greater integration between clinic guided research, as it were, empowering patients to provide us with data with the appropriate regulatory framework that we can then leverage to understand further, I think would drive us faster further than currently, with a much more separate world of research and clinical translation.
Thank you for that. We just have one more minute, so I hate to leave some questions out, because there's a really great question about maintaining relevance and driving adoption when working with diverse stakeholders. I just wonder, though, Ernest, if you could give us this future directions, what's your vision for the critical next steps in transitioning from this nice to have to must to have technologies in ALS and beyond?
Yeah, maybe I'll echo Alvaro's comments just now. I think the more, obviously, the three top things we need are more data, and more data, right? But this idea of trying to link together different kinds of data collection, I think, is really critical. Getting better access, making more public data sets so that the whole community can work together to develop the AI tools. And then I also believe that in all of these observational studies, we should try to collect biofluids as well, and being able to connect, because there's going to be a limit to what we can do with just symptomology. We really have to get beyond that and find the connections between the clinical and the molecular.
Well, that wraps us up with this section. I'm sorry we didn't have time for more questions. We can take them through the chat or email afterwards. And thank you, Indu and the team and all of the presenters, for the opportunity to moderate this session.
Perfect. Thank you so much, Carl. And I know we're running about 15 minutes late. We're going to try to, hurry up here. So thank you for the panel. And the next section that, we want to discuss very quickly is that developing AI tools in ALS. So I'm going to invite my colleague, Steffi Hans, Natalia Lucina, and Sean Peno to come in and kind of, talk to you guys very briefly about what we are developing and the excitement that we feel we want to share very quickly. Steffi?
All right, well, Natalia is sharing. Sean, as the actual expert, as PALS and Pathfinder, go for it.
Yeah, my name is Sean Pennell. I've been diagnosed in September of 2023. I've recently been asked to become part of the Pathfinders group here with the EverythingALS, where we try to fill the gap in our community between the professional experts and the individual disease experience, with a mission to empower our experiences and knowledge to the community. We're now being accessed through the newly launched EverythingALS app, and we can now, we also provide community touch points with our bi-weekly and alternating fireside chats that we have online. As you can see with the slides here, we have hosted on our YouTube channel. They've garnered nearly 500,000 views across 50 different countries. And this global reach does highlight the clear demand for accessible, detailed information on ALS research. Feedback from the entire ALS community has defined this combined approach of care with citizen research, paving the way with this new industry approach. Next slide. EverythingALS has recognized that citizen research are at the forefront of driving innovation. Here are some examples of the latest expert talk where PALS asked 36 very detailed scientific questions. Our goal is to remove the bias that traditionally sidelines patients from that input. PALS are the most invested in advancing ALS research, and their questions can lead to breakthroughs and should be considered equally in the research process. As an example, my wife and I often find ourselves doing our own research, reading complex scientific papers, struggling to understand them because they're not that easily accessible to us non-scientific community. We frequently have to search for definitions of those scientific terms just to follow the content and reread them over and over again. As a segue, now EverythingALS has launched a chatbot to help with that kind of thing. The success of the expert talks and all the PALS questions has inspired EverythingALS to develop a chatbot that is limited to the information collected in the EverythingALS expert talk series. This limitation is important as it concentrates those responses and increases the trust in that information. More importantly, the chatbot provides an intuitive, centralized platform where we can access that information from our experts in one place, fastly and easily digestible form. This particular chatbot can direct users to relevant talks, help with scientific sections, and answer complex queries. I've even used it to Google certain things like what our last doctor was talking about with meals. What's the best meal plan? You can put that into the search chatbot, and it gives you some excellent resources. So with that, I'll pass it back to Natalia.
Thank you, Sean. Yeah, we definitely were very inspired by what we saw in the chatbot and the type of questions which were asked in expert talks. And not surprisingly, the similar problems are observed in the research community as well. This is the actually picture which OpenAI created for us to represent the field of ALS research, basically showing that there is plenty of unstandardized data available. And unfortunately, researchers often spend a lot of time going through spreadsheets, trying to standardize the data available from the past historic data in order to move the innovation further. And based on, we also saw the same problem when we actually started doing our own research as a part of student ambassador program. We wanted to create the overview of ALS field and ALS clinical trials And we started with a small piece of information, only analyzing the data from clinicaltrials.gov across the past 3 years. And even that, as you can see on this slide, took a ton of hours for us in order to get all the data from clinicaltrial.gov, pull the spreadsheet, try to create additional filters in order to standardize it, and then pull additional insights on what is the different type of clinical endpoints used in those past clinical trials, and what is the use of digital health technologies, and what are the trends in this research. And we envisioned and tried to create a chatbot on this small data set. And we were impressed how much time savings we can get if we had that tool available for all of the researchers and patient community. And we also demoed this kind of demo of the tool during our summit, and we got an amazing feedback from all of the research community and pharma industry and clinicians. And that brought us to the next step and our further vision, which Steffi would share.
Right. So now that we have a proof of concept, vetted with all the end users, why not go at scale? That's our modus operandi at EverythingALS. So we are right now starting in collaboration with Amazon Web Services to go at scale. The goal is to create an intuitive AI-powered solution that can handle larger data sets from multiple sources to bring insights to all end users. That means the researchers, the PALs, the CALs, industry, really with the goal to start with enhancing clinical trial design, but also accelerating innovation in ALS research. And why stop there? This can go a lot further. Other diseases have exactly the same problem. This is nothing but a tool and ultimately a playbook that can be repeated over and over again. So that's a little snippet update on how AI in the grassroots of patients and the EverythingALS team develops little valuable tools, and turning over to our next section.
Thank you. Thank you, Steffi. And we'll move to the next section here, which is the big tech and ALS, generative AI and AI for good. So we are going to, I want to invite our moderator, Dow Jensen. And I've known Dow for probably, gosh, we're going to, 20 years, I think. Dow was the founder and CEO of Oak Rocket, and she merged with a bigger company, Campus UOL. Now she's the president of the Campus UOL, and which does AWS business globally. We are very fortunate to be partnering with Compass and with a cloud support for us and also all our partners. So we actually provide, I think, modalities part of it, and many other companies who work with us. And Dow's expertise in the tech industry has made her sought out speaker at global events. She also serves as the Young Presidents Organization president in Pacific West chapter. With that, I would love to invite Dow Jensen.
Thank you, Indu, for having me, and thank you everyone for joining today. Today we're going to talk about big tech and why we need big tech and Gen AI and for the good of science. Today we have Kelly Norell with me from IBM. She has been involved in many projects and events. Her role is in advancing AI-driven solutions for healthcare, particularly in areas of depression and sleep disorders, Parkinson's disease, ALS, metabolic diseases, where they can use speech analysis as a diagnostic tool. Kelly has highlighted their collaboration with research institutions, foundations, pharma, and showcasing different successful collaborative models. She can also address the challenges of scaling AI, focusing across country-specific regulations, and IBM's approaches to the use of in-house systems like LLMs for compliance with healthcare regulations, rather than relying on external platforms like OpenAI. Welcome, Kelly.
Thank you very much for the introduction, and thank you, Indu, for inviting me to talk here.
Jeff will be our next speaker as well on the panel. He's a vice president of Salesforce and ALS caregiver. Jeff will share Salesforce's initiatives in supporting ALS through programs by Brooke Eby, such as the internal Slack channel ALS Force, which has, I love that name, by the way, Christian, which has over 800 members ready to engage in ALS-related efforts. He will also highlight the ALS's Together community and other initiatives leveraging Salesforce's technology and resources to raise awareness and drive engagement and support in the ALS research care. Welcome, Christian.
Yeah. Jeff, you mean?
You
Yeah.
got it. Dow, thanks very much for having me. No worries. Indu and Christian, thanks a ton. And Jen DiMartino, I know you're out there. Jen, we love you. So thanks for having me here today. Appreciate it.
Apologies.
No worries. Easy.
My page jumped to another page again.
It's all good. It's all good.
And our last panelist is Luca Fascini. She's president and CEO of Sage BioNetworks. Luca will highlight Sage's BioNetworks efforts in promoting open science and creating open digital health data sets to foster collaboration in biomedical research. He will emphasize the need for standardization in digital health, particularly in areas like mobility, accelerometry, and share insights from the experience with disease detection using wearable data. Luca will also discuss the importance of open innovation and the challenges posed by proprietary data and the lack of shared tools for frameworks for validating the new devices. Welcome, Luca.
Thank you, everyone. Thanks for having me.
So I do have to have a hard stop at 9:30, unfortunately. So I will be handling this over to Christian. That's probably why his name was, and to Jeff over, because I do need to jump off briefly over to Christian. But let's start with a good question of me having been in a lot of big companies like Symantec, Oracle, and why it's important for our big tech companies to get involved for data. So you've shared examples of organizations that are advancing AI for good in healthcare. Can you highlight any of the panelists, specific collaborations that have driven the most positive outcomes or broader adoption, and how can these be applied to ALS research? Kelly, Luca, or Jeff, if you'd like to take that.
Kelly, do you want to go first?
Yeah, sure. So it's interesting. Many years ago, I was also working for DREAM. That's a crowdsource for science that's run by Sage now. So it all starts there, as also Ernest was saying before. We need large amount of data, and you need to collaborate. No one has all the answers. No one has all the information. So, for example, in our projects with the different organizations, being academics, being industry, being nonprofit, in a sense, we get some data, we get some insights from the experts on the field, and then we provide like the AI. And I think in several of the areas we're showing we can do great things together. But again, we need the left and the right hand to wash the hands.
Kelly, do you have a few slides you want to bring up? And yeah, why don't we do that?
I do.
Yeah.
Mm-hmm. Yeah.
Picture says 1,000 times better than the words.
I totally agree with you, Indu. I'm so visual that it's helpful to be able to have everyone on the screen and not just pictures or audio.
Okay. I think I don't have permission to share.
Oh, now you do.
No, I don't.
Yeah.
And we can see it, Kelly.
Yeah, but are you seeing the right screen though?
Yes. Yeah. Advancing healthcare through innovation at IBM.
Perfect. Fantastic. So, in a sense, is what James Berry was mentioning before. Aren't we familiar with this situation? Can you tell me how do you feel? Can you tell me how do you speak? Right? We know there is a lot of bias in how a person will answer a question. There is self-report bias. There is, depending on the mood, depending on so many things. So what we are trying to do at Digital Health at IBM is precisely avoid these type of issues. we want to capture the data without asking directly, answer me to this survey, like the LSFRS-R, for example. So we use speech as a window to the mind. And why we do this is easy to collect, is scalable, is low cost, and we extract both linguistic and acoustic markers. That is, what do you say, how you say it, and also signals from the voice. We have projects that deal with neurological conditions, with psychiatric disorders. We even can tell you if you are ingesting, let's say, MDMA, ecstasy at a high or low dose based on you telling me a story. And we also have work in metabolic disease, chronic pain, and others. So I will show a couple of examples of what we do. So first of all, we can quantify different aspects of the mental or general status of a person, being emotion, stress, anxiety, things like psychosis, cognitive status, motor dysfunctions, Parkinson's disease, and ALS. We have studies on pain, and we map based on naturalistic speech to inventories or surveys. So a couple of examples to show like the different flavors or things we do. So for example, the top paper mentioned here is we have data from English speaking countries like the US. We have data from Mandarin in China. If you can imagine, the language is very different, the culture is very different. But when we compare the markers for psychosis, they are maintained. That's, I think, is remarkable. I mean, it's very interesting. This other study that was done with Pfizer, we wanted to see if we can detect, again, using speech, if the person is in the on or off state of levodopa. So the person will take levodopa after a couple of hours. The levodopa stops working. That time is different for every person and also depends on what you ate. So can you detect that in an easy to find way? And it has been known in the community that when they compare people with Parkinson's to people that are controlled, people with Parkinson's use less action words. So I thought, okay, that's interesting, but probably when they say, what did you do this weekend? A control will say, I went for a swim, I play soccer. And the person with Parkinson's says, I watched a movie, I kept reading. So we wanted to test if this is true or not. So we give patients in the on or off state with levodopa, and when they are in the on state, they are closer to controls. To describe two pictures, one in the on state and one in the off state, and then we switch. So half the population receive each picture first, so it's not related to the picture itself. And we found that effectively people in the on state with the medication, they use more action words. So that is showing that how can you move your body and how do you feel is influencing cognition and the words you use to describe an image. Then, for example, in interviews of narratives, tell me about your life, we can detect the cells of agency in either controls or people with psychosis. And psychosis don't have a sense of agency. That is, they, in their perception, their voluntary actions don't have an influence in the world. And if you think living with that is pretty hard, right? Then we also have another study that we analyze Reddit posts for people with discussing different types of mental health. We were focusing on suicidality, and just based on this data-driven approach, we are able to recover something called high top. That is a new method to classify mental disorders that is replacing the DSM-5 and the ICD-10. And again, all these data-driven with open-ended questions. Then we work with the Answer ALS that was also mentioned here before, and analyzing the data from their app that is tracing and tapping with the finger, and then speech tasks, read some sentences, describe a picture. We are able to infer the LSFRS total, but that's a little bit surprising, but that's because we have also inclusion of the finger tracing and tapping, and also the LSFRS are vulvar and speech with very good performance. Then with the EverythingALS, we were trying to infer the ECAS, and that's a metric to measure cognitive impairment. Is between 35 or 50% of the people with ALS will have some cognitive impairment, and about 10 to 15% will have FTD. And unfortunately, people that have both FTD and ALS have a much worse prognosis. They die sooner. So it's beneficial if you can detect early and change how the person will be taken care of. And then recently, also working with data from EverythingALS, we were able to classify not only controls versus patients. We designate pre-manifest people with ALS that they themselves think their speech has not been affected, with controls that they have normal speech, meaning that we can point to early detection. So hopefully with the early detection, hopefully soon, there will be some interventions to be made. And I don't know if you guys have questions now or after the end of the panel.
Yeah, we'll do it end of the panel if you have any questions. Thank you.
And Jeff, coming from Salesforce with a lot of data sharing, right? How do you feel the role of, app and open data and innovation play into our efforts, or Kelly or Luca?
Yeah, so first of all, I really appreciate being here. My name is Jeff Green. I've met a fair amount of you in Boston and in around the space. My story with ALS, just briefly, and then we'll talk about Salesforce because I think it's important to set the scene. So I lost my father to ALS in late April, after about, I'd say, a 3 or 4 year fight, which included Jen DiMartino, beloved Jen DiMartino, who's on the phone or on the video. We transferred my dad's care to Merit, and ALS ONE was very helpful in doing that and took a leadership role in that, which really, I think, prolonged my father's life for the care that he got up in Boston and just the spirit. And so in front of a world of people, it was Jen that was kind of the fulcrum in that. So Jen, thank you. On behalf of my family, thank you. But I would say that there's an interesting story behind how I got to Jen. I work at Salesforce. I've been there 9 years. I'm a former federal official in housing and lending, in disaster housing and lending, in particular, hurricane work. So I come with a little bit of a disaster background, but my boss's boss's boss is Jen's boss at ALS ONE. Mark Sullivan is the board chair. So I had this sort of weird moment in time where I found my way to Mark. My father was having a very good experience with TUDCA. It was odd. It just, every time he was about to start slurring his words, every time he took that, it just kind of helped him sort of straighten up a little bit. And being New Yorkers at heart, once you're a New Yorker, you're never not. He wanted to let everybody know, and there was no place to sort of let everybody know the experience he was having. So I went to Mark, and we got to Jen. Jen helped my dad get to Merit. But we also started down this path of, can we create a single place where everybody with ALS who's either a patient, a caregiver, or a doctor can be on one platform? And this sort of began the idea for what ALS Together has become. And as the early days started, Brooke Eby, who many of you know Brooke is an employee at Salesforce. And by the way, we had started ALS Force, an affinity group inside the company to focus on this, to focus on what we were doing with ALS. And sort of the emergence of Brooke came, and it was just, this woman is just like, it's not just that she's a social media phenomena. She's a leader and she's smart. And she grabbed the CEO of Slack, Denise Dresser, and just said, we've got to build this community. And it never would have gotten done without Brooke. And I'd say we're about 6 weeks ago, it went live. And why is that? You know, when I listen to the incredible work that IBM's doing, it's incredible, right? The Salesforce thing is a little bit different in terms of creating an initiative where everybody, and remember, we're a global company. So think about that for a minute, where everybody who has ALS is a caregiver of someone with ALS or someone who has interest in that community can be in one instance of Slack, sharing information, being together, and being the gateway for whatever might be necessary. I'm a huge Indu Navar fan. And so when Indu thinks about and her brain gets around, well, what if everybody's in one place? What can I do? What can I effectuate as a step 2 or a level 2 or a double click if I have access to everybody who's in one place? The ideas for that are exponential of where that might go. And so the ALS Together community, which I think we just went live with caregivers maybe 48 hours ago or so, is the beginning of a centralized place for where everybody with ALS can be. And whatever initiatives, whatever things might come from that, start from everybody being in one place. And that's sort of like, I'd say, the focal point for Salesforce. I think the second thing that I'd like to highlight that was almost a reach back to the previous session, which I think is very, very important to realize, is that the pace of AI inside big technology companies is breathtaking. And so think about this for a minute. We're a company of 80,000 people. Go look us up. If you don't know what Salesforce does, we're the best customer experience people in the world. We're the best at this. This is what we do. And we're sort of tying AI and autonomous AI 24/7, 365, a digital worker, a digital agent on your wing for whatever you're doing, who's working and supporting your work while you're asleep. This notion of autonomous AI and digital agents on the wing of a jobs to be done framework, whatever those jobs are to be done, from mindless tasks that require no human intervention to somewhere in the middle, to somewhere where it requires as much help as possible. 8 weeks ago, we completely turned the focus of our company to autonomous agents and an agent force approach. That's 8 weeks ago. Salesforce is not the same company it was 8 weeks ago. That's how rapidly this pacing of innovation is taking place. So what does that mean for all of the blessed humans on this call who've dedicated their careers in one way or the other, or all in, on this ALS fight? It's fascinating. Where is all the data? How much of that data is in one place? What's the tooling and the tool set in a trusted configuration with guardrails at the core? And remember, guardrails are a huge part of where AI is going, at least in our company. And then what does that mean? We could get on this call a year from now and just be nowhere near the same. I saw a stat I just took a screenshot of, doing something used to take 24 hours, do 10 minutes. Just wait, month over month, the pacing of which autonomous AI and that ability to extract data for utility is going to move at a pace that is going to be shocking. And then the question is the community in a right place? Is there coherence across that community in order to take these tools that are moving at such a rapid pace, focus them in the right places to have the right breakthroughs to help people?
Thank you, Jeff. Like you said, there's a lot of traction and things are changing companies and people's experiences with AI so much now, and some of the challenges that are going up. Luca, because of all your collaboration work that you've been working on with your company, could you talk about some of the challenges you've encountered when working with stakeholders that you've seen occur in your business, in terms of everyone working together between the healthcare providers, pharma, and patients?
Absolutely. Well, first of all, thanks for inviting me. It's a pleasure and an honor to be here. I lead Sage Bio Network. That is not a large company. We have 110 employees. So if you square that number, you still don't get to the 80,000 of Salesforce. But we pack a punch because we want to be the glue. We want to be the glue among all the actors that are working in these fields and trying to advance the understanding of disease like ALS. As it was mentioned in the previous panel, there's really a market failure in bringing data to who needs data, right? Like data is produced in isolation, sometimes by researcher that have a primary research scope, sometimes by healthcare provider that has a cure objective, and it's never thought to be shared and reused downstream by those actors. And then on the other side of the spectrum, there are people that would love to have this data, life science, digital health companies, startups, medical device, citizen scientists, and those struggle finding the data because no one has built a Google for data yet. That's as simple as that, right? Really, we struggle to search and find health data to be working with, even when this data is open. And the definition of open can really vary. So what we do at Sage is really we try to solve this problem. We're trying to connect these two sides of the world, the data creators and contributors with data users. And we do so in a variety of ways. And I want to, if I'm able to share a couple of things, we build tools to make that easy. We run a very large database called Synapse. I don't have visual feedback. Can someone tell me if you see my screen?
We see it.
Yeah.
Cool. So we run a very large database called Synapse with more than 3 petabytes of data across 20,000 projects, 6,000 active users. These are the data sets that are the most downloaded. These data sets come from researchers, mostly NIH researchers that are now mandated to share data, but we know that sharing does not imply reuse. Again, you can share as much as you want, but if people don't find you, it's like you never shared. And that's to your question of what is one of the challenge. Well, one of the challenges is really that we're trying to build restaurants and get people to eat at our restaurants without publishing a menu. Like nobody's gonna come. So by creating social signals on this data, what are the most downloaded data sets in this database? I'm not gonna go screen all the 20,000 of them. I just want to see what the community is looking at because that's where I might be interested in. And obviously, a big piece of it is search. So we built, of course, an LLM. We threw an LLM on top of our Synapse search. So if you now log into Synapse, you can chat with Synapse and ask questions about this 20,000 data sets that are, openly accessible, but hard to find and hard to understand if it's what you need. Do you have any data about ALS? And the chatbot will say, yes, we have this 4 or 5 things. Is this what you're looking for? And then you can go on and ask more questions about that. Well, is there digital health data in there? Is there transcriptomic data? How many patients? And that way you can hone in what, as a user, you might be looking for. We're also building another resource that I think is really cool, and we launched less than 6 months ago. That's called OpenChallenges.io. Again, our mission is to bring more eyeballs to the data that's already there. It's fantastic that there are folks that are focused on creating more data. We absolutely want that, but there's a lot of data already that's not being seen. So that's our focus, is to make that data seen. And one way of making that data seen that, in fact, we've learned through our experience, like if you go to Synapse and you look at those most top downloaded data sets last month, in position 4 and 5, you see BRATS. What's BRATS? Well, BRATS is a data challenge. It's not a data set. I mean, the data set is the data sets that's used for a data hackathon. And that's why it gets so downloaded, because it has become some sort of benchmark in the field. This is about brain tumor segmentation. So we learn that hackathon, which means data plus a task on top of that, are really effective in creating engagement across community and bridging across community, bridging the science community that creates the data with the computational and AI. I am an AI researcher myself, that could develop algorithms to mine and solve tasks on that data. So Challenges have another very similar problem to data sets. They're hard to find. There is no Google for challenges. So we build this public service that's called openchallenges.io. You can go and say, I want challenges. What challenges have been run in the past about ALS? And what it pops up is something that we and the DREAM community that was mentioned earlier by Kelly. It's a challenge from, I think, almost 10 years ago about stratifying patients. So you had to develop an algorithm. As a participant of this challenge, you have to develop an algorithm that, given some biomarkers of ALS patient, can understand, can predict progression. And the way that algorithm is tested is in a blinded way. So there is a piece of the data sets that is hidden, and the algorithm is developed in a piece of data set that is public, and then the algorithms is tested on the hidden one. Yeah, these things are very successful. One of our best example is a challenge that we ran for breast cancer prediction based on mammograms. It was very successful because there was a big prize for it, and that's a very important thing to the question that was asked earlier, like what are the big challenges in this world? Well, one of the big challenges is that the incentives are not aligned. But if you bring citizen scientists, computational community around the table, you give them a prize for the one that builds the best algorithm to detect breast cancer for mammograms. Turns out that challenge was run in 2017. Turns out that 6 years later, 2 of the top 3 winners of the challenge got FDA approval for their algorithm. They started companies and they got FDA approval for their algorithm. So that's really, to me, a fantastic translational outcome. So that's what Sage tries to do. We build search engine on data, we build challenges, and we build data portals, which are places where all data around the same theme gets put together. So you can create some data gravity for a community to build around. This is the last thing I'm gonna say. There is an effort accelerating partnership in medicine, AMP ALS. I'm sure you all talked about a lot here. That's being spearheaded. We are a big part of that. That will be a place where a lot of ALS data sets are collected. And to conclude, one very close to my heart theme is digital health, disjointed from a specific condition. It's really close to my heart because before taking over Sage a couple of years ago, I was an entrepreneur. I started a company called Evidation Health, took it from 4 to 300 employees, 5 million users in the United States, trying to use data from wearables and smartphone to direct patients to the best possible option for care and research, so connection to clinical trials. As part of that work, we've developed algorithms to tell you when you're getting sick based on your data from your Fitbit, Apple Watch, and Garmin. And we found that these things are really hard to validate. And in fact, we wrote a piece on the Lancet Digital that calls out for better reproducibility in the digital health world. Why is reproducibility hard? Well, because all the 3 pillars that David Dono at Stanford requires for frictionless reproducibility, which is open data, open source, and a culture of benchmark, none of those is available in digital health. Most of the data is proprietary because it comes from proprietary devices, cannot be easily shared. Tools, therefore, are not being created and shared publicly. And finally, you cannot create leaderboard. You cannot understand what tools are the best because there are not tools out there. So to conclude, if you want to bring the world of biomedical sciences closer to the pace of progress that we see in AI that was just mentioned, where you cannot possibly publish a new chatbot and say it's better than GPT-4 because people will drag you in a thing that's called the chatbot arena, which is a way of testing your solution against all the other best one, right? If you can't build this culture of frictionless reproducibility where software is open and could be easily benchmarked against everything that's out there, we are not going to have the pace of progress that we're seeing in AI. And so that's what we're trying to do at Sage. I hope you follow us at sagebionetworks.org.
Thank you, Luca. And I know we are going to do a quick Q&A, so I'm going to give probably a minute or two for you to answer. As you're talking about, what a good segue, you said frictionless technology. Seems like technology is not, you know, a showstopper anymore because we can have a lot of cloud data and AI. What do you think about the friction in adoption when you're working with different stakeholders like researchers, healthcare providers, pharma, patients? Where do you think they are culturally to adopting, you know, what you're building? I mean, you know, which is fantastic.
I'm a techie. I come from the tech world, and there's a lot of things we don't do well in tech, and we all know which one they are. You know, addicting teenagers to Instagram. That's one of those, right? But I think the pace of innovation there has it's a world that has less constraints in terms of risk aversion, which could backfire, as we just described, but could also leave openness for trying out half-baked solution, have them out in the open without what's called the nirvana fallacy, without the fear that if it's not perfect, it shouldn't be seen. So I see that I see that maybe life science and you know the healthcare industry in general does not have that culture of let's put something out that is half-baked and let someone else take that on, because sometimes that could be risky, right? Like sometimes that could really hurt patients, so for good reasons. But I think there are sub places like closer to the spectrum of research and further away from care where the risk is higher, when we could be more, we could be daring more in terms of like, put things out for other to build on, and don't be too worried about people calling out your flaws. Your flaws are going to be someone else's gain. I think that culture is not that present in life sciences as much yet.
Yeah, good point. Good point. And the same, you know, kind of thinking, Jeff, what do you think? I mean, like, as you guys are coming out with Agent Force at Salesforce, and, you know, we are very fortunate to have all of you guys to be, you know, thinking about ALS and bringing all this technology. What do you think the challenges are for adoption? And what do you guys are doing in general, not just in ALS, to actually address this?
Yeah, well, I think first and foremost inside the company, I think as a core values-driven company, this notion that we're going to rally around things that are important to employees and run with that is never going to change in what we do. So the ability to spin up an affinity group and have, you know, a whole focus, you know, ELT, executive leadership team focus around ALS inside a company is very rare. And we have that, and that's not really going to go away. So I think inside the company, it has to be core values driven. I think that one thing I'll say, though, having spent a lot of time here, you know, and being in other software companies, and I know Indu, it's where you and I see eye to eye, is, and it's why I'm going to spend a lot of time personally on Vision 2030 with everything ALS, is I think when you get to the boardrooms of some of these private sector companies and you say, here's the contribution that we need you to make in the ALS community writ large, there has to be a framework and a plan around where these companies feel like they can make an investment and feel good. Not, you know, of course, an in-kind contribution and person power in the form of hackathons or technology is incredible, but also a monetary contribution. And it's what's drawn me to the Vision 2030 piece, because I see the framework around how private sector companies can see where a contribution is going to go and yield benefit in a broader community. And so I think that's very important. I think there's no daylight or no mistaking about how the ALS community is working in synchronicity to make improvements in people's lives and hopefully find its way towards a cure. I would say, last comment I would say too, is I think I speak, and I know Indu, you're in this category too, of a group of people who have lost somebody to ALS that will simply never stop fighting and never not make a contribution towards outcomes in this space. And I think it's something that I don't know about other communities that fight disease or work together to fight disease if they have that same, replication in the constituent base, but I will never, ever stop. And so that's something as we think about those decade-long plans, God willing, or however long those plans take, is the ability to count on people never leaving the community until the outcomes are hit. And again, I would say I've seen a lot of CEOs, I've been with a lot of CEOs, I've been with a lot of big companies. They want to hear that and see that in people's eyes as well too. Are you in to finish this thing off? And I think that's something about the ALS community that catches, I know, our leadership team differently than others because of the passion that the employees have and that the people who care about the community do. So long-winded answer, but
thank you, Jeff.
Yeah, you bet.
And having this force, really, ALS force, ALS agent, or sorry, force agent. We are very fortunate. And I have one last question as we wrap up to Kelly. Kelly, as you're doing, you've been working with us at IBM, and also, you're working with many other organizations in ALS and other, other companies other places where you have to get the data because, what you're doing is really very, very important, and your time and energy, what IBM is putting together with a lot of resources there. What do you think about data sharing and data standardization? I mean, do you have any thoughts about you can share with us how we can do better, where we are? Would love to hear.
Sure. So again, from experience we got when we were working with the DREAM challenges, first of all, and this is what Christian is sharing, thank you, Christian, and also something that Luca was saying, you don't need to be perfect. You don't have to, first of all, know everybody can be the best, because it's very hard to be, if everybody is above average, we are in a weird place, right? When you aggregate solutions and when you aggregate data, you get for sure better results. That's why the crowdsourcing approach works so well. And yes, you need, I remember when we were doing that first ALS challenge with data from PROACT, that those were the failed clinical trials for ALS. It's interesting because at one point we realized data coming from Europe was using the normal metrics for us, and data coming from the US were using imperial metrics, and people didn't realize that. So something as simple as using the same metric system, right, was important for the challenge. So yeah, you need to be able to, again, my biases are different from other people's bias. So the minute that you put more minds working together, for sure you will get better results.
Thank you, Kelly. Really appreciate all of you taking the time. And I know we talked about Price for Life as all of you work. And, we do make sure that we collect all the information, and we actually acquired the patents from Price for Life because we didn't want all the effort that was, wasted, because a lot of good things they had done and when they were closing it down. So we will continue to, like Jeff said, we're here to see the end of it. So with that, we really feel like very fortunate to have this army. And I would love to invite our next session with Lee, and brain-computer interface and discussion and Q&A. And I'm going to invite Dr. Lee Hochberg, who is our host here, to take it over.
Great. Thanks so much. Good afternoon on the East Coast and further east, and good morning out west, and happy Friday. And thanks so much to Everything ALS and to ALS ONE, to Indu for the kind introduction, to Jen, Christian, Sarita, and so many others for hosting this great 3-day symposium and today's Neurotech Day. This is actually a perfect segue to go from AI to BCI, and not just because of the perfect alphabetic progression, which we'll just give Indu credit for. That was perfect. So this session is going to be about implantable BCIs in particular, which are devices that can record from the brain, that can decode brain activity, and that can do something useful with it. And I'll be quick without any slides. Specifically, as we've learned over the past few years, for people with ALS, IBCIs, as you may hear them called throughout the session, have allowed people with ALS to regain intuitive control over computer cursors, control over external robotics, and more recently, to have direct intended to audible speech applications. And today we've assembled in what's really a rapidly growing field from both industry and academia, a great group of clinicians and scientists in this rapidly growing field. It turns out that everybody on the call here are good friends today. And this group in rapid progression are going to highlight just a little bit of either the most recently published data or the most recently revealed data across ongoing trials in implantable BCIs and or from devices that are on their way there. And I'm just going to direct everybody to the program guide for the background of the speakers, and we'll take off shortly. So I'll give you just a brief overview on everybody. We're going to hear first from Kurt Hagstrom at Synchron and Romina Najad from Neuralink, who are going to tell us a bit about their industry-based clinical trials. They're going to be followed by Nick Ramsey in Utrecht and Sergey Stavisky from the BrainGate Group at University of California, Davis, who are going to summarize their recent reports in the New England Journal of Medicine. We're then going to hear from Ben Rappaport at Precision Neuroscience, Matt Engel at Paradromics, and Rob Franklin at BlackRock, who will provide updates on their implantable BCIs. And then last speaker will be Jessica Kellerman, who's going to introduce the recently launched Implantable Brain-Computer Interface Collaborative Community, the IBCICC. And then hopefully at the end, we'll have a few moments for Q&A from everyone. So with just that brief introduction, I'm going to hand right over to Kurt Hagstrom.
Hi, everyone. Can you hear me okay?
Yeah, we sure can, Kurt.
Wonderful. Let me how to share my screen. And let me know if you can see the presentation.
You're good.
Wonderful. All right, everyone. Well, thank you so much for having me. I'm going to talk to you about a company, an industry company that's developing an implantable brain-computer interface. And this is a really evolving technology. As Lee mentioned, there's a lot of, I think, great collaboration on the industry side of it to bring this technology to light. I will be talking to you about an investigational device. So this is under investigation from an FDA standpoint. My name, again, is Kurt Hagstrom. I'm the Chief Commercial Officer at Synchron, about a biomedical engineer by trade. So we're not commercializing, so we are developing the technology. So today, I'd like to talk to you a little bit about the principles in which we look at this technology and how we're developing it. Next, I'll talk a little bit about the procedure, how the device functions, and how we've now translated it into the clinical arena. I'll talk about the two trials that we've done to date, and then a little bit about what's next for us in BCI. So I always start with this in most of my talks because I feel like I ground myself. This is Graham, one of our first patient in our switch trial in Australia. He's one of, I think over 14 million people today living globally that have some form of paralysis where you have an ADL deficit. One of the things that I do like to show, if you can see it, Graham wrote this book about his experience with a BCI, with the BCI, and is when he's going through his journey getting the BCI. So it's really great, and I'll pass this on to the community afterwards. But this was an important moment for us where, again, it was that first human experience with the technology. When we look at what grounds us, it's really around these three elements: independence, autonomy, and privacy. I think it's been mentioned quite a few times today already. The element of autonomy for individuals is and independence is almost a human right to that degree. Making sure that people can get back some level of independence. And I think we're coming at a world where there's so much discussion around technology that it makes a lot of sense for us as you look at these implantable BCIs coming in a world where innovation and technology is moving so fast. So I think you're going to see a really amazing merge between these technologies. And then lastly, privacy, I think, is something that was also talked about privacy of data and the ethical use of that data. So again, that's really critical and something that we put on the forefront of our development, ensuring that we maintain that element of privacy in patient with patients' data as we move forward on our journey. All of this does not happen. We don't sit in our lab and try to design this by ourselves, and we're not developing for people. We're really developing with people, with some amazing advocacy groups on the left-hand side there that are really ground us every day. Although we're trying to, obviously there's a support to find a cure for ALS, but there's also living with ALS, and that's what I think we're all about. We've also gone over 250 people from various stakeholders, from managing physicians, therapists, to more importantly, caregivers and patients, people that are living with ALS, that we understand their needs, because everyone's a little bit different. And so when we sum up, again, looking at our principles of independence and autonomy and privacy and what this system, the design of the system needs to do, it's really around 5 core things. And that's number 1, immediate use. So it works in the first session. It's got to be easy to learn, so low cognitive effort. It can't be just, an hour or 2 a day. This has to be used for 24/7. It has to be used and be able to access for 24 hours. You need to be able to use this all day long and when you want to use it. Calibration's also something that's really critical, and I think important as we look at this technology moving forward, that has to really either have an element of self-calibration or extremely low need for calibration as you move forward with the patient. And I think that's also part of that access, 24-hour access, and make sure that it's there when you need it. And then lastly, stability. Stability is something that every individual's there's a progression of disease, and it's critical that there's a stability of function and performance over time. And I think we've been able to see that with our technology, which and again is really important, that the performance element of the system, as you look at an implantable prosthetic like this, that it has that stability over time. When we frame the utility of this, we look at activities of daily living, whether the basic of feeding, bathing, banking, or instrumental activities. I think we all know that again, this digital world has now taken over. Almost 100% of people have a cell phone. 78% of people do banking online. 46% of consumers use telehealth. Right, that's really moved forward in the healthcare arena. And then you look at e-commerce, there's no question that's a massive field. So our life has gone digital, and I think we're at a good point to capitalize on that. So when you think about an individual's functional independence as it relates to these activities of daily living, I think more is being published and understood around this. Is really kind of 9 domains that we see that can be digitized, from communication to management of health to entertainment to productivity. This functional independence allows for personal control of one's environment, information, and choices. And so these are some examples of Mark, who I'll talk a little bit more about, but he is one of our patients in our latest trial, the COMMAND trial, doing different tasks within the study, within the COMMAND study. So next I'll talk a little bit about the procedure. So this is an endovascular procedure. So this is where you access the venous system, and so there's no craniotomy. You access the venous system, and you place the BCI, called the stentrode, into the blood vessel. The novelty is that there's a rich history of clinical knowledge around putting technologies like this in blood vessels. And if you look at the coronary space, there's decades of experience putting things in blood vessels, whether a pacemaker lead or whether it's a stent to keep open an artery. And even in the neurospace, there's a lot of technology that's been there. So what you saw there is the placement of it within the vein. This is underneath the skin, that the lead comes down, and then you have a transceiver there that collects the data and then transmits the data out for utility. As I mentioned, the stability aspect of this approach, this is again, the vasculature on the left-hand side. And then you can see the stentrode on the right. And this essentially starts to embed within that blood vessel, allowing for that stability over time. So it heals within the blood vessel, but without actually having to touch the brain, which is kind of the novelty of our approach. When you think of well, how does this system work? Think of it as you have motor outputs when you're motor intent and you think about moving your hands. With this technology, there's an element of restoration. That's why they call these motor neuroprosthetics. So you think about moving just different gestures. So moving your foot or your hands, then that signal gets transmitted via the BCI. And then it gets, again, transmitted to another unit here called the signal processing unit. And that translates the digital motor output into digital action. And so that's what will end up controlling the end use devices, whether it's a smart device, computer, potentially robotics in the future, and also the environment. So now I'll talk a little bit about the clinical translation of brain-computer interfaces. And I do want to mention, because Lee, you're kind of kicking things off and hosting this session. None of this clinical translation would have happened without decades of amazing science that has created a foundation of knowledge in relation to translating neural signals and understanding what those neural signals can do. So it's been amazing work that we're lucky at Synchron to be able to capitalize on this knowledge. And there was a lot of talk about sharing information. And I think this field of implantable BCIs really epitomizes that. I think it's been in a tremendous, we have a collaborative community that has started. But again, I think just the science and academic nature of the implantable BCIs has really been there from the beginning. And I think it continues to be very collaborative. And I think that's needed from a data perspective. So when you look at our clinical pathway, so back in 2019, we did our switch trial. This was in Australia. We did 4 patients, and this is really our first in human study. Next, we wrapped up we started another study here in the United States, the FDA IDE study. This was started in 2022, and this was, again, 6 patients. Most of all the patients within the switch and command trial had ALS. There was one that had brainstem stroke. And next, we're moving forward with our Empower trial, and that's hopefully getting kicked off next year. And that will have our second generation system that I'll talk about in a moment. But let's talk a little bit about that first switch trial. So as I mentioned, this is our first in human trial. There was 4 patients, all had ALS. The primary endpoint of this study, so again, what we were mainly focused on was the safety element of this procedure and implant. The data was published in JAMA Neurology in 2009, so it is something that you can look up. This was followed for 12 months, and oftentimes these implantables, you have to make sure that they have that long-term durability. So 12 months was that mark for the primary endpoint, and they've gone well beyond that, obviously 12 months at this point. So there was no serious adverse events, no negative lifestyle changes, and these systems were used independently at home. They also had no again, downfall of their activities of daily living, only improvements on those, whether it's texting, emails, or online shopping. So for us, this was really a testament for us to move forward and continue our journey in this clinical translation of this technology. So now we're wrapping up our command trial. So we've done that 12-month follow-up. This was a great collaboration with folks at Mount Sinai, University of Buffalo, UPMC, Carnegie Mellon, and it was funded by the NIH. So we first of all, thank you all from the primary investigators, and they were great partners with this. And we also thank all the patients that were part of this, those 6 patients. So when you look at some of the learnings from this trial that just wrapped up, this was the first FDA-approved IDE trial for permanently implanted BCI. So it's very exciting that you see this first really long-term transition of this implantable technology. So it's exciting for the field in general. We had excellent safety results. So no SAEs, serious adverse events related to the stentrode, which is that portion you saw in the blood vessel, the brain, or the vasculature. It was easy and accurate to deliver. So again, the beauty of this system is that the techniques needed, the surgical procedural techniques are standardized within any kind of cath lab environment. For those doctors that are treating stroke every day, these are the same doctors have the same skills that can place the stentrode device and also the receiving device. So again, this is not something new from a technique standpoint, which is really good. So again, this is a fully internalized system. Again, it was safe and accurate surgical procedure, as I mentioned. And again, there was good use. All of these, we worked with all the patients in their own homes. So I'll talk a little bit about some of that experience within the clinical trial. So this is the first, this is Mark, and he's an amazing individual. So he came to the trial, and he's upper limb impairment. This is the first moment where we activate the BCI. So for him, we often see this really emotional moment where they know as a disease progresses that he has something there to be able to connect to, I think, really at the end of the day, connect to everything, from a digital standpoint. When we worked with Mark, we focus around this on the SMART goals. So what would you like to do? He wanted to do entertainment. He wanted to control the Amazon Fire tablet. He wanted to be able to set alarms and things for medication, as well as more entertainment. He wanted to watch TV and use the Fire tablet for that. So in this environment, in the command trial, we did these types of tasks and make sure that you can connect to these big tech platforms and devices. And I think that was talked about before, that I think the partnership with big tech is really important. And I think, how do you seamlessly connect to these things? And then one of the things we're working with some of the big tech partners is around this concept of BCI hid for Bluetooth. So having a very unique connection between technology that is not based on your fingers or a mouse, left click, right click, but it's based on your neural control. And I think there's a unique opportunity there, using your neural control to completely change how we think about that digital interaction, replacing, being able to use hand gestures, replacing typing, replacing the voice if needed. And so I get really excited. And I think I really want to also thank the big tech players. Their accessibility teams are really amazing, and they share very much the same vision and mission that we do at Synchron. So looking ahead, as I mentioned, in 2025, we hope to kick off our next trial. That is going to be with our second gen system called Sync One. This system is completely implantable. It's Bluetooth. It does connect to the cloud where needed. But this is I think will be a very exciting moment for us. This is what will be our commercial system that we'll be able to move forward with into pivotal. So lastly, wanted to put this information. This is our Synchron. This is a link to our Synchron BCI community. This is where you can go on and learn a little bit more about what we're doing, as far as what the BCI does, how it works, what are some of the clinical results, as well as this is a place where everyone will understand when that next trial is up and running, when the FDA has approved the protocol, and we can get things kicked off. And so if you would like to join, feel free to do that, or you can reach out to me specifically. I put my email here, so you can learn a little bit more about what we're doing here at Synchron. So thank you so much. I appreciate the time, and back to you, Ellie.
Wonderful. Kurt, thanks so much. Sounds like great, both introduction and update to everything you're doing there at Synchron. I will now, seeing myself, yeah, okay, good. I'll now hand over to Romina. I think I saw you a little earlier there. Romina Najad.
Yep. Hi, Lee.
Oh, hey, how are you? It's all yours.
All right, great. Let me try to get this shared. All right, awesome. That's a really exciting talk you just gave, Kurt. I'm excited about your command trial and the results that you guys have, released. My name is Romina Najad. I'm a clinical trial manager at Neuralink, and today I'm going to give you an overview of our PRIME study, as well as some of the updates on our participants. So just to begin, our mission at Neuralink broadly is to create a generalized brain interface to restore autonomy to those with unmet medical needs. And in order to achieve this mission, we've developed a technology stack that we have here. So I'll start with the left image here, which is showing our N1 implant, also referred to as telepathy or link. You can see this implant, it's about 25 millimeters in diameter. It's the size of a quarter, and kind of protruding from underneath the implant, you'll see our flexible thin film array, which consists of our threads. These are 64 threads with 16 electrodes on each thread. This results in a total of 1,024 channels. We use a surgical robot to implant these threads because the threads are, each thread is thinner than a human hair. And so even the most skilled neurosurgeon cannot implant these threads in the brain. And so we developed a robot that we use that very carefully inserts these threads in human brains. And the robot itself, you know, is really a technological marvel. It's in and of itself, in that it reduces the implantation time for the thread insertions. It precisely maps the insertions of the threads with brain motion as a result of breathing and the anesthetic regimen that our participants receive. And it also avoids blood vasculature. And so kind of once, you know, participants are implanted with our N1 implant, we are able to decode that neural activity from their motor cortex into a useful output for them. So in the PRIME study, we're focusing on a digital output such as cursor and keyboard control. So what does PRIME stand for? It stands for Precise Robotically Implanted Brain-Computer Interface. This is our first early feasibility study at Neuralink, and it's currently ongoing. We launched this study last September, and today we've enrolled and implanted 2 participants. And the goal of this study is that, you know, again, as I mentioned, we're going to use our N1 implant to allow users with paralysis due to spinal cord injury or ALS to control a computer cursor or keyboard, only using their thoughts alone. And one thing to mention here is that our implant is completely implanted underneath the scalp, and so it's cosmetically invisible to others. So no one will really know you even have this implant implanted, and it is wirelessly. It works wirelessly with the computer itself. So you can see here in this little schematic of the participant, you know, it's we're decoding that neural activity into some useful output. And for this trial, it is ongoing, so we're still collecting data. The primary endpoint is 12 months following implantation. Our first participant, Nolan, who is pretty well known at this point, has reached around 9 months in his journey, and so we're waiting for him to reach his primary endpoint. And following that, participants will be followed up for up to 5 years, and they can continue using their device and working with our BCI engineers and team all along that study duration as well. So what are our near-term objectives? I mentioned the overarching company goal, but for our near term, we're focusing on progressing on our early feasibility study, with our primary objective being to demonstrate the initial safety of our robotic surgery and the implant itself. And then our second objective is to improve our product from learning from participants. And so we have you know, a wide array of preclinical data to support the launch of this early feasibility study. But now that we're implanting in humans, we can actually use their feedback. They can vocalize their feedback or communicate with us via other assistive technology and to give us, you know, what would work for them. And so we really work with them on a tailored approach to unlock certain functionalities that each one requires. And I'll kind of get to that with the differences in our first and second participant. So this is an overview, just briefly, of our surgical procedure. So you could see what the PRIME study involves is we make a skin incision, and then we make a craniectomy. Again, it's the size of about a quarter, right over your motor cortex. We do a directomy as well, so we remove that extra membrane above the brain, and then we use our robot to insert the threads. We slide the implant in place, and then the skin comes over top of that implant, and then it's cosmetically invisible. This is a video of just our robot in general. It's a pretty big robot. It weighs about 1 ton, but I just wanted to show you how thin our threads are actually. So you can see those are red blood cells. We have the threads at the bottom of the schematic, and then we have the robot needle in and of itself, and then the blue is that dura mater, that membrane that is kind of over the brain. And so you can see how thin these threads are, but even thinner is the robot needle that kind of picks up these threads and inserts them into the brain. This is a craniogram footage of one of our participants. And so you can see here on the left side, we have that 25 millimeter-ish burr hole you can see that the exposed surface of the brain and those white circles are the targets that we place using our robot. And then you can see how the targets are placed precisely, to avoid blood vessels, not only major blood vessels, but even the smallest blood vessels that we can see. The way we target is interesting. We use functional MRI imaging from our participants when they're under the scanner, and then we look at, we use neuronavigation to target areas where we see the highest functional activity when they're thinking of motor tasks, such as moving their hands, moving their wrists, moving their fingers. Obviously, they're paralyzed and they cannot actually do that, but we can still map that activity using functional scans. And then on the right side, you can see this is post-thread insertions, and so you can see kind of what that looks like on the surface of the brain. You can see very, very little bleeding, if any, on this scan. And so, I just wanted to show kind of what that looks like for folks. All right. So once we implant the N1 implant, so what happens? As I mentioned, we're decoding neural activity that's streaming from the brain wirelessly to the computer. All of that decoding happens on the computer itself, and then we model that decoded behavior into some useful output. So in this case, it's the grid game task, moving the computer cursor to select boxes on a grid. And then now I'm going to go into our actual participants. P1, also known as Noland, who's has stuff out there if you're curious to see. But here are some of the feature points that I want to highlight. So on the left, you can see Nolan, prior to being implanted, his main assistive technology was using a mouth stick. And so you can see that he's able to play chess using his mouth stick. This has a bunch of ergonomic and other limitations for him in that, after a couple hours or a certain amount of time when he's using this mouth stick, his neck muscles tire out. And he also needs his caregiver to set up this iPad for him so that he can actually use it and engage with the computer in this way. Sometimes even like if he pushes too hard on that iPad, it'll move out of the way, and then he has no way of like shifting it back. And so it's largely dependent on having someone with him while he uses his current assistive technology. And then on the right side here, you can see a couple different images. One at the top is Nolan playing Mario Kart using his N1 implant. So this is post-implantation. He was able to actually play this game, which he wasn't ever able to do since his injury. And so that was really special for us to unlock that for him. And then at the bottom schematic, this is a live stream of him playing Civ. He's pretty active on X and posts, updates about how he's doing and how he's finding, the device and how useful it is for him. And then thirdly, I just wanted to point out that this video of him on a plane somewhere, in which Neuralink was not even involved in this at all. So this is not like a planned demo or anything, but Nolan is sitting there, using his, wireless Neuralink implant to create cat memes and share that with the world. And so we thought that was pretty cool. All right, just to go a little bit into some of his BCI performance. So on day one when he began using his implant, Nolan actually broke the previous human world record and achieved a 4.6 BPS. And essentially what that means is he was able to move the cursor at a specific speed to hit the select targets. And then you can see at, several months after, he's still continuing to improve, not only in precision, but accuracy. And he can now select even more precise targets on a bigger grid and has achieved about 9.5 BPS. And just BPS, I think in general is a complex measure, but for those who are not super familiar, you can actually play this game on Neuralink. We have a link down there, WebGrid, and see how fast you can actually do this. So the average Neuralinker, I would say, is able to achieve about 10 BPS using the computer mouse, and Nolan is right around 9.5 right now. And so we're really unlocking capabilities that we're hoping to continue to improve on, but they're reaching almost able-bodied level performance just by using his BCI. And then another thing I wanted to highlight is just weekly hours of independent BCI use, right? So we use, all of our BCI sessions are done in participant homes, but outside of these sessions, we like to track what is the number of independent hours of use that participants are actually using the device? So when they're not involved in a Neuralink session, is the device actually useful for them? Are they able to use it to achieve their goals? And one way we track that is by the number of hours that they're independently using the device. And here you can see you have hours on the y-axis and date on the x-axis. You can see Nolan is averaging, like 50 hours-ish a week of independent use of his device. And so he's very happy with his device and continuing to use it. Although I do want to preface that, we have a low N and, it's an early feasibility study, but this is just data that we're pretty excited about. And for P2, okay, so P2 is a very special person as well. He has a spinal cord injury that he sustained from a car crash. And essentially for him, he has different goals, like as any two people may in the real world. And he really likes to design things using CAD software. So this is him using his N1 implant to actually design things on fusion software. He was unable to previously do this following his injury using his assistive technology. So this was a feature that was very important for him to be able to do. And here he's designing kind of a mount for his N1 charger using his BCI. And here you can see on this right side image, I don't know if you can see my cursor, but at the bottom here, that's the mount that he 3D printed and used, and he designed that using his implant. And then this is a quote from him: taking an idea, putting it as a design, and actually having a physical item as a finished product makes me feel like I'm building things again. And so, he can't use his hands to build, but at least he's able to get back to what he loves to do using this BCI technology. And then this is a video of P2 playing Counter-Strike. I do want to preface that our goal is not to fully replace the assistive technology participants use. And so in this instance, we've actually combined the BCI implant with his quad stick. And so he's able to have two joysticks now where he's able to aim using the BCI implant, but he actually moves using his quad stick. And so he's using those in parallel here to be able to play this game that he couldn't play previously with just the quad stick alone. So you can see here, he's, hopefully killing all the bad guys here, but that's him playing Counter-Strike. And then for P2, we see that the weekly hours of BCI use are less than our first participant. And mainly this is what we think is due to the differences in their general lifestyles. So P2 really likes to be outdoors, and he spends a lot of time on the farm and kind of in the studio building and designing things, which is different than, what P1's lifestyle is. And so he's still tracking and increasing that weekly hours of use over time, but just want to state that there are differences between people and how how often they do actually use their device. And that's about it. If you are curious to learn more about our study, you can join our patient registry, or you can learn more about it on this web link down here. We also have this patient registry launched in Canada and UK as well. Yeah, that's the end of my talk.
Wonderful. Romina, thanks so much for that great update on the work that you're all doing there. We are now going to fly, not quite as far as we could, but Romina, I'm guessing you're in California at the moment or somewhere near the West Coast. We're now going to fly to Utrecht and hand over to Nick Ramsey, who I guess I don't know, Nick, that you're actually in Utrecht at the moment, but you can tell us and provide us an update on the work from the Utrecht neuroprosthesis.
Thanks, Lee. Thanks for inviting me. This is really an amazing event. I'll see if I can share my screen. Okay. So I'll be talking with a little bit different perspective. All right. So I'm going to talk about research that started about 12 years ago. And I just want to show the team first, because in the end, people forget. There's a really nice team that works on all the different facets of implanting a BCI in a person with ALS, in a late stage ALS. And they do all the work that I have the privilege of talking about, because I don't program anymore. So the general idea we had earlier than 2012 was that, and Lee already published this paper with the UTA array in a patient, and our idea was, well, what can we do to actually move BCI or test it, whether it's worth what we think it can be. And we decided in 2008 that we would try to simply get a click in a person who was locked in, because we believe that people who are locked in have severe problems with communication, are the ones that stand to really benefit from the technology, provided the technology can actually offer them what the minimum requirements that they would have. So we decided to work with locked in persons, and most of the people that we encounter are people in late stage ALS. We would work with a fully implanted BCI because the primary goal was to prove that it works at home without assistance of us as researchers, which, you know, would be the real proof of utility of BCI for people with disabilities. So our measure of success, we thought at the time, was to measure the number of hours or number of hours in a day that a person would use it. And I'll get into that a little bit more. So we chose a locked-in person as locked-in people as our target population, fully implanted BCI, which was only possible because we worked together with Medtronic. They had a DBS device that they, for different reasons, were equipping with sensing technology, which gave us 4 tiny amplifiers in a fully implantable DBS system, but we had different electrodes, as I'll show in a second. And then we decided to put all the effort into getting a super reliable brain click. And as Synchron just showed, with the brain click and scanning software, you can really get quite far. And mind you, these people have no means of communicating by themselves. So autonomy is near zero and privacy is completely zero, because everything they need to do is mediated by a caregiver. So that's the thing that we were hoping to be able to assist in. So the device looks like this. So there's some electrodes that go through burr holes, which are small drill holes in the skull. We slide them underneath, we fix them in place. Then there's a wire underneath the skin that goes to the device here. That's about the size of a Zippo lighter. That device lasted 5 years, 24/7, because of some ingenious technology by the Medtronic people. You have this plastic antenna that you kind of fix to the t-shirt and a wire, and it goes straight into a Surface Pro, a simple computer. So we were looking for people who were interested, and we met Hanneke. Her name can be mentioned because she was in the first publication that we had in 2016. And before we implanted her, we talked to her. So, So we decided to work with people with ALS who have decided to have a ventilator implanted because we didn't want to run the risk of having someone go into surgery and not be able to come out without being dependent on a respirator. So that limited the number of, the target population that we could work with, but there are a considerable number of people that actually decide to have a ventilator and to live on. In those cases, of course, they're pretty much always locked in. So this is a picture of the New England Journal, the medical journal that decided to accept our story. And they made this drawing. So there's a few electrode grids. They're plastic strips that we slide them between the dura and the brain, basically along the inside of the skull. Each has 4 electrodes. And then there's wires that go underneath the skin, and we fix them here. And again, like I showed you, this is the setup. There's the antenna, there's a little box from Medtronic to kind of handle the signal, and then it goes straight into a not too powerful laptop or tablet, which does all the work, like translating the brain signals into instructions and running the software. In this case, it was Tobii software that you used for daily use. So this is an X-ray after the surgery. These are the electrodes, these dots here. These are just connectors, and then at the bottom here, you can see the device that was amplifying the signal and sending it out wirelessly to that plastic antenna. So Hanako was 58 years old. She's locked in. She had ALS since 2008 at the time, 2015. And we implanted her with this device in 2015. Mind you, the amplifier, this is, it was designed in 2009. Half of it is still DBS, which we switched off, which is electrical stimulator for Parkinson's. We switched that off. The other half was recording. So we only had four amplifiers. So we can only connect 2 of these 4 leads. And we chose the leads that were on top of the hand area. So the way we decode the signal is quite similar to how Synchron decodes the signal. The performance is similar in certain ways, but the electrodes are in a very different place, of course. These are directly on top of the part of the brain that controls movements of the hand. So basically what happens is the person is at rest, then we ask the person to try to move the hand. But of course, nothing is happening because she's completely paralyzed. But when you try to move the hand, the brain basically does the same as when you actually move the hand. And the signal basically becomes more noisy. There's more fluctuations here. And then when you stop moving, it goes back to rest. What we get from the brain, I just leave out the mathematics and stuff. That's not interesting here. But this is the signal that we get out of the brain. We call it the control signal. That goes up when the person moves the hand. And it keeps going up until it hits a threshold. At that moment, we collect evidence, and this is hundreds of milliseconds, until we are completely convinced that the person is trying to move the hand, and then we generate a click in the software that the person uses for daily interactions. So just to show you how this works, this is an early display of how she uses it in her Tobii speller. She still had to get a little bit used to it, but just to give you a good feel of what the system looked like to her and 3 more patients we implanted later. But this story is mostly about her because she's been in the study for such a long time. So this is very standard scanning software. You have to click when it's highlighted. When you do that, it will break up. Now it goes from top to bottom. When it breaks up and goes from left to right, though. It'll just take a second to do this. Now, the important thing here is how fast the highlight moves. So that's basically the scan rate. And the faster she can generate a click, the shorter the scan rate. So for eye trackers or for people with switches, it can be faster, but she started with 2.5 seconds between items. Now, so she can basically generate a good click within 2.5 seconds, but the software, of course, has to go through all kinds of items that she's not interested in, which causes her to be able to make 2 or 3 clicks per minute, which is very low, but it worked for her. And then we started interacting and we listened to her and learned from her. She's basically a co-researcher in the study. And she told us what she needs. And one was a cold caregiver. So she could click through menus, but when she had some care need that she wanted to communicate, she didn't want to go back and, go through all the menus. So she had an escape signal. And the way we did it was we asked her to move her fingers continuously, basically identifying a long click, which we can distinguish from the short click. And when the long click is generated, the software flies back to this menu on the right, where she can click on caregiver, which gets a small alarm, and then she'll get attention and she can explain either with Tobii and icons with pre-programmed text, or by eye blinks or other communication, what she wants. The second thing that really made a difference is at night. When she lost the ability to control the eye tracker, she lost the ability to call a caregiver at night, and she got very restless with this, very unhappy. So she asked us to be able to switch it on at night, and we did. But it turns out the brain signal in anyone, when people fall asleep, becomes very, very noisy. So our click decoder started to go crazy. We had like 200 non-intended clicks per hour. So we had to go back to the drawing board and we found out that because the brain is so noisy, it basically activates any decoder that we could come up with. So we ended up with a situation where she would listen to the ventilator, which clicks every 4 seconds, and she would generate a click every 8 seconds. And she would do that reliably. And then through trial and error, we found out that we got nearly 100% accuracy only after she would do this for about 80 seconds. So we thought she would never use that, but she was very happy with this. And the reason that she was happy that she got it to work and so reliable is because when the night alarm goes off, the caregivers get very upset because they're supposed to sleep during the night unless there's a need. But if there's false alarms, very quickly they will not switch it on at night. So we had to prevent that, and we succeeded quite well. And this is what she said when asked about what she thought of the system.
Nick, just to let you know, we're not hearing that audio. So if you wanted us to hear it too, you could reshare with the sound on.
All right. It doesn't matter. It's translated anyway, so. All right. So she spoke Dutch. I don't think many people speak Dutch, but she's very happy with the system, and she's been asked many times by journalists through email, and she regarded it as her system. And I want to take just a few seconds to go through this slide. This is the end, the complete story. The first paper about her got published in the New England Journal, probably because we showed that she was using it without us there, a certain number of hours per day. And that was basically in the first period. Now on the y-axis, you see the number of hours per day that it's used. We don't know what she's doing. Talking about privacy, like Britt, and she didn't want us to know or be able to reconstruct what she was doing with the device. We asked her kindly. So we only know the creation and the closure of log files that were otherwise empty. And so we could accurately track how much she was using it. And you can see in the beginning, the first year, she was using it not that much. It's only outside because the eye tracker failed to work outside and on holidays. But then at some point, she started using it more and more. And in hindsight, we identified the different stages that we encountered. So in the beginning, she could use the eye gaze device very well. You see that at the top, it's black, until it started to falter and became more and more erratic to use. And at the same time, her eye blinks started to fail. And at that time, she started to rely on this system more and more, and quite rapidly, because at that point, she didn't use the eye tracker anymore. At this point, we managed to have the night decoder working. And then she showed up to basically using it at 24 hours a day because the other hours is disconnected for washing and things like that. And she used it for a solid 2 years, most of the time. At some point, the night decoder didn't work very well, and we don't quite know why, but we couldn't get it to work, so it was switched off at night. And then here at the end of year 7, her performance went down very fast, and that was correlated with the last twitch that she could create. So overall, I think this is very nice proof that ABCI can work even as simple as this, because she could still accomplish things that she could not do in any other way. And of course, we're all looking for more sophisticated devices, but we'll see what kind of sophistication we need. Anyway, from her and some other participants, we learned a couple of things: that there's significant clinical benefit even at this level, because it offers something that the person cannot accomplish in any other way, which is privacy, autonomy, and dignity is an important part of it. The whole system was completely invisible, as a matter of fact. The signals turned out to be super stable because they're fairly large electrodes on top. We didn't have to calibrate. She didn't have to calibrate. She could do it herself. And I think we kind of checked every 6 months or so, but there were barely any changes needed, except for close to the end where the whole signal started to deteriorate. Quite affordable in the sense that it's a fairly simple system. The battery lasted for 5 years. It's no longer in production, but for the study, that was a good finding. The things that we learned is that the BCI decoders absolutely fail at night. Anything that works during the day is going to have to be completely redesigned at night. It's possible, but it's still a huge challenge because 80 seconds is a bit long. We found that the brain signals decline in amplitude and quality in people with ALS. And in some persons quicker than in other persons, but it does mean that it's inevitable that at some point in the progression of the disease, the device will not work anymore. And it's fairly close to when communication, any remaining communication, is also no longer possible. And it, like eye blinks, where you depend on a caregiver. And the third thing we found is that click speed varies across individuals. Some people are able to generate a attempted click within a second, but people that are a little further in the disease seem to have lost the ability to acutely generate this attempted movement. They can't imagine it anymore. And then that slows down the system. We had one person where it took 6 seconds to click, and another person where it took 8 seconds to click. So it really depends on the stage of the disease, whether the system is going to be snappy and accurate or not. And I want to close this conversation, presentation with one of the 3 clinical pilot studies that we're running in Utrecht. In this one, we're collaborating with Johns Hopkins. It's called the Corticom study. It's open for people to subscribe. Instead of a long URL, just type Hopkins and Corticom at the bottom in Google or any web browser. You will find the website to get more information. And the big change here is we're going to look at we are looking at 128 contacts instead of the 8 that we had in the study I showed you. And we're looking to decode some basic elements of speech. You'll hear a very nice story from Sergey about speech a little bit later. I do want to point out, I always point out that the people that we work with do no longer speak, so they don't have any sensory feedback that goes back into the brain that helps with decoding. So we kind of figured that we have to adjust our expectations and hope that people that no longer move late stage will be able to generate a couple of command words so that they can control a cursor by attempted speech. Anyway, with this, I want to close, and I want to thank you very much for giving me the time to talk about this.
Excellent. Thanks very much, Nick, for that update from Utrecht. We will now fly back west to hear from Sergey Stavisky at the University of California, Davis, talk about some recently reported results, also in the same journal as the report that Nick was just describing.
Great. All right. Thanks, everyone. It's a privilege to be here and to tell you about our work. So I think with this audience, I don't need to explain how important it is to restore the ability to communicate for people who are unable to speak. And I'm going to be telling you about this story that, as Lee just said, was published in that same issue, and there was also a bunch of press around it. And similar to in Nick's talk, our participant and his family have agreed to speak with the media and have their video and quotes out there. So that's why you'll be seeing them. Otherwise, of course, we would respect their privacy. Just to introduce our group, BrainGate is an academic clinical trial. Lee Hochberg is actually our sponsor investigator, so he's our fearless leader. And we have 4 sites across the US. Like in the previous academic talk, we do not build implantable hardware. We use what is already available to kind of push the envelope in terms of the neuroscience, the medicine, and the algorithms, and really demonstrate what can be done with this type of technology, hoping that industry will follow and operationalize it, make it slicker. So a speech neuroprosthesis is in principle very simple. The idea is a person tries to talk. We record their brain activity that would normally move the muscles, but in this case, they are mostly paralyzed due to ALS or spinal cord injury or a stroke. We run those data through a computer algorithm that uses artificial intelligence to figure out what it is that those brain signals correspond to. And in this case, words appear on the other side. So let me play a video that illustrates how this works. This is showing a participant named Casey. He is a man living with ALS. He's in his 40s. And you'll hear his voice. It's severely dysarthric. Outside of his spouse and one assistant, basically no one can understand him. he's going to be trying to read these random sentences that we're presenting to him at the top of the screen. And as he tries to speak, you'll see the neurally decoded words appearing at the bottom. So that's not decoded from movements or voice activity, that's just from brain activity. And at the end of each sentence, he's going to use an eye tracker to look at the done button. He can also use a neural cursor like Romina showed, and it's going to speak it out loud. So let me play the video.
Maybe after we're not in school anymore. And they're very dedicated to that.
To briefly explain how this works, we start with recording intracortical signals. So we're putting in these so-called Utah arrays manufactured by BlackRock Neurotech. So we're recording from a couple of hundred individual neurons in areas of the brain that normally control speech. These data are then sent out to a computer. For now, this is a percutaneous wired system. As you've heard, there are lots of companies trying to get rid of these wires, which is amazing. That brain activity is first processed by a type of artificial intelligence algorithm that predicts the phonemes, so the units of speech that the person's trying to say. And then a couple other algorithms then take those phoneme probabilities and convert them into words and sentences. And so at the end, you get a complete sentence on the screen. And then when he selects that he wants to speak it, that is read out loud in a voice that actually sounds like him, basically like a deep fake of his voice using some previous recordings he's made. It was previously mentioned that it's important for this to work as quickly as possible. And so we were fortunate to get a video of that very first session. So this was a few weeks after his surgery. He's spent at this point 30 minutes trying to say sentences and record the data. And this is that first time we're going to turn it on, and you're going to see him and his family's reaction. It's a really special moment. So let me play this video too. So you can see that those first two words appeared, and he just stopped, and everyone was crying with joy. And we had to actually pause the task until we, high-fived and composed ourselves, and then kept going. So with some additional data collection, the accuracy became very good. So it plateaued at about 98% accurate. So he can say any word in the English dictionary and most proper nouns, and he can add words to this dictionary. And 1 out of 50 words is incorrect. So extremely accurate. Of course, he doesn't want to use this to copy sentences that we present to him so we can measure the accuracy. He wants to talk to people. And so I'm going to play a video on the second day of use. And this is the first time we let him say whatever he wanted with the system.
Hello, is this on? I'm looking for cheetah.
That was referring to his daughter who was wearing a cheetah onesie.
Sweet butter of mine.
So you get the sense of how important that is to him to actually be able to speak to his very young daughter for the first time in her memory. We've since refined the user interface. So this video is a few months later. The voice will sound better, and you'll see that he can indicate with his eye tracker or cursor when he wants it to speak it out loud. So this is like in those previous talks, we're learning from him how he actually wants to use it. It's important that he can select exactly when it vocalizes so he doesn't interrupt people or vice versa. And you also see that when he self-reports if the sentence is not 100% correct, the language model presents some additional options, which usually has the right sentence in there. You'll also notice he's learned at this point, he doesn't actually have to vocalize. He's basically just sort of slightly moving his lips.
Hello, my sweet child. Would you like to talk with me?
He likes this a lot. It is literally an order of magnitude faster than his best alternative, which is a head mouse or one of 2 people in the world that can understand him. In the New England Journal of Medicine paper, we reported the data I'm showing now. So he's used it then for almost a year, for over 300 hours. He talks with his family. He resumed working full-time. He Zooms, he Slacks, he writes emails. He talks to his friends. His social life has become much richer. But what's really exciting is it was working so well that we wrote to FDA and we requested permission to train his care partner, his spouse, to put the system on and off, so the little electronics box that goes on the head, and we automated the software. And after that happened, you can see this usage just completely take off. So he's using it 50 to 80 hours a week, basically from the time he wakes up to the time he goes to bed. And he's liking it a great deal. And one thing that we found amazing is that the performance has actually increased. So the accuracy is sustained and his speaking rate has increased. He basically, when he got ALS, he worked with SLPs to learn to speak more slowly so he could be understood for as long as possible. And now he's unlearned that and realized that he can try to speak faster, at least to the speed he can with his ALS, and the system still understands him. So there's this increase in communication rate. And just to close out, he is much more eloquent than I am, so I'm going to let him explain why this is important.
People who do not have the ability to communicate well or quickly are often isolated and lonely. Having something like this will give them hope and opportunity to be more of themselves. I hope that we are at a time when everyone who is like me have the same opportunity as I do to have a device like this that will help them communicate. Let's all make that happen, okay?
So yes, I think a good call to arms for the many people in this session working on these type of technologies. So just to summarize, we've shown what we call a brain-to-text speech BCI because it ultimately decodes text. There's future work trying to restore prosody, tone, pitch, things like that. It's very accurate. It uses that at home. But as in similar talks, important caveats, this is one man with ALS. He is still able to move to some degree and to vocalize a little bit. So there's potential concerns down the road. We will, unfortunately, probably find out if he continues to use this. It's a big team effort. I want to specifically acknowledge Nick Card, the postdoc who led this project, and my lab co-director, David Brandman, the neurosurgeon co-directing our group. BrainGate is a big team, and a lot of people made this work. So I guess we'll do questions at the end. But if you want to learn more, braingate.org has all sorts of information about our clinical trial, how you can potentially enroll, other research that we're doing across this wider team. Thank you.
Awesome. Thanks so much, Sergey, for that great presentation and an update as well since the paper was published. We will now, having hopped over the East Coast a few times, we will now actually land on the East Coast, and I'll turn over to Ben Rappaport at Precision Neuro.
Great. Thank you so much for having me. Give me just a second to share my screen here. It's a real pleasure and an honor to be here with this group of friends and colleagues, and to be in this important annual meeting. Thanks for having us back. And it's amazing to see how rapidly things are moving in this field, and to feel like something many of us have been working on for a long time, and many people and families have been hoping for. Really, I think, gaining speed and traction for the ALS community and for the spinal cord injury community and for many other communities who stand to benefit. Thanks everybody for your work, and thanks for giving us an opportunity to share today. I'm going to spend a little bit of time presenting some of the recent work that we've been doing at Precision Neuroscience on implantable brain-computer interfaces, and this relates to ALS and a number of other allied conditions. Are my slides coming through okay?
Yeah, I see the first slide. I thought
Good. Okay. Yep. Great. Just want to make sure that I'm so I'm Chief Science Officer of Precision Neuroscience. I'm also Assistant Professor of Neurosurgery at Mount Sinai in New York City. And I just want to start off so we don't give the thanks short shrift by thanking so many of the people involved in this work, most notably the first 25 patients who've been involved in our early clinical studies, and hopefully we'll get to touch a little bit on that, as well as to the trainees, the patients and families in the audience today, an amazing team at Precision, and our clinical collaborators across a number of institutions. I'm going to try to move kind of quickly, and a lot of the background content has been already presented in some of the other talks. But we're building implantable brain-computer interfaces, which we view as really communication devices that translate neural signals, biological neural signals, into digital bit streams, and then use artificial intelligence to translate those digital bit streams into intelligible forms of information, whether it be speech and language or motor output, to enable people living with paralysis to interact in a natural and smooth way with the digital world. And we view this as having tremendous potential across a variety of disease states. And of course, as many of us know, this builds on many decades of work, represented by a number of the people here in this panel. And I won't go over it other than just to say that all of us stand on the shoulders of many thousands or more of researchers and individuals with lived experience that's allowed us to collectively build the field to where it is today, really on the precipice of doing tremendous good in the context of hopefully not just promising clinical studies, but I think soon we all hope becoming part of the standard of care. At Precision, we really think about brain-computer interfaces as communication links between the brain and the digital world. And in thinking about them in that way, we view the bandwidth of the communication link as being tremendously important. And there's a number of ways of thinking about that bandwidth. Many of us just use electrode number as a kind of proxy for that bandwidth. Not perfect, but nevertheless a good first approximation, at least at the hardware level. And so we've worked hard to try to make the interface that we use, which I'll show you a little bit more about in a minute, as scalable as possible to number of electrodes in a way that spans as much functional cortical surface area as possible to enable the brain to have a smooth, high bandwidth interface with the digital world. And even though that's sort of just a one-dimensional way of looking at the interface, it of course, scaling in that way requires innovation across a number of different areas, and that includes microfabrication, storage, and advanced processing of massive data sets that's enabled by advances in artificial intelligence and digital signal processing, miniaturization, customization of implantable electronics and wireless technology. And as these technologies have, over the last several years, entered the clinical domain, the importance of interacting with the regulatory agencies has become more and more critical. And thankfully, I think we have a good community now with a nuanced, interaction with the FDA. And I think one of the presentations at the end of this session will touch on the collaborative community, which has been a real, I think, benefit to all of us who are presenting this afternoon. Many of us know, and especially I think I love presenting after Sergey. It's just so inspiring, and I feel like it's our duty to make the work that teams like the UC Davis team have shown to be possible. In initial promising results, to make that scalable and standardized and part of the standard of care. But I think we all share a sense that with bandwidth of the communication link comes the ability to communicate in a sort of smooth and effortless, natural way that Sergey was showing in the prior talk. And certainly we aspire to doing that at Precision, maybe even to change the nature of digital communication, at large. And this was hinted at, I think, in in the previous session where the folks at Salesforce were talking about how, and also Kurt in his presentation on Synchron was thinking about, what does our interaction with the digital world look like in the years to come as brain-computer interfaces, enter the real world. I'm just spend just a moment thinking, just to address the way we think about the interface at a sort of basic neuroscience level. Certainly when I trained, there was a dogma around the need for intracortical electrodes as the only way of interfacing in a highly reliable way, in a decodable way with the brain. And we've sort of tried to move into a different paradigm, a complementary paradigm, through which, rather than interacting at a one-to-one neuron or clusters of neurons level, we think about our interfaces interacting rather with at the scale of cortical columns. These groups of physiologically and functionally connected tens of thousands of neurons that fire and wire together and represent information in a kind of cohesive way across the cortex. This is a preserved architectural element across the cortex, whether it's visual or motor or otherwise, slightly different in different regions, but nevertheless quite similar. And when we think about that way of interacting with the brain, it's possible to sort of think about things in a two-dimensional grid rather than a kind of depth probe context, and that has many advantages. One of which I'll just sort of show here to give some intuition, which is that what you're seeing here on the screen, this is, hope it comes through without being too choppy, but this is slowed down about 100-fold, but each of these dots represents the voltage, instantaneous voltage from one of 1,024 microelectrodes on the brain surface, in the right frontal lobe. The hot colors, the reds and oranges, represent relatively high voltage. The blues and purples represent cool colors, and this is the nature of the data that we obtain from the brain surface, in real time from a thin film electrode array that we use that coats the brain surface rather than penetrating into the brain. And maybe a few years ago, it was thought that the ability to decode from such an interface was perhaps limited, but we and others have shown that actually this data is very, very rich, highly decodable, and offers a number of advantages in the context of an implant. And so this is what the device looks like. In the middle, you see our thin film array, which is about 22 microns in thickness. It's modular, so we've used many modules at a time. But this one module contains 1,024 electrodes. The small ones optimized for recording are 50 microns in diameter, and they have the same spacing as the well-known Utah electrode array with its 400 micron interelectrode spacing, but it spans a larger cortical surface area. And we've used multiple of these together, and we've also worked to develop a minimally invasive insertion mechanism, which you can see shown here in a schematic way, where we implant the electrodes into the subdural space through what we call a cranial microslit insertion technique. So these are soft, thin film electrodes, but nevertheless, through a minimally invasive cranial aperture, we can place them under the dura directly onto the brain surface using image guidance, and using kind of on the fly GPU accelerated compute. We can do all kinds of using the sort of grid arrangement of the electrodes because we preserve the spatial because we preserve the
Let's see. It may just be me, Ben. I lost your audio and your video, and you are frozen, which increases the likelihood that you don't hear me telling you that. Just to make sure it's not me, either Jen or Indu, could I ask you just to say hi and let us know?
Yeah, I'm not hearing either. Do you hear me, Lee?
Yep, I sure do. Okay, and reappearing. We'll give him just a moment, and hopefully he'll be able to come back on. Well, one possibility is that we may, in a moment, shift. So Matt, I'll ask you to be prepared to take over, and then we can return to Ben if need be. Let's see. I'm looking on my screen just to see if Matt's logged, or sorry, if Ben is logged back in.
Sorry about that.
He's back.
I lost
All right.
power for a sec.
All right.
Can you hear me again?
Yeah, you're good. Go ahead, Ben.
Share.
This, as Ben is doing this, this is the point when my Zoom doesn't work, is when I say something along the lines of, when you hear a conference about brain-computer interfaces, you really hope at the very least that Zoom does what it's supposed to. Right, that's the, so.
Yeah, I'll claim responsibility for that one. I didn't realize that my battery was running low, but thank you, Lee, for graciously saving me there. So I think I was just trying to emphasize that the spatial arrangement of the electrodes allows us to do certain things on the fly, and to use the physiology and correlate that with anatomic proximity of the electrodes to use spatial features and not just the temporal features of individual neurons. And there's a huge amount of rich information content that contributes to decodable features when you view the electrode information in this way. We've done a fair amount of work showing the relative information content of adjacent electrodes, just again sort of questioning the trying to probe into where is the information content in what spatial and frequency bands. And we've done some work also on microstimulation, so thinking towards the future of how do we do the right side of the BCI, and that's a good subject for another time. Thinking about how to make these into scalable devices that work in the real world as part of medical standard of care. We've tried to, as many of you are familiar with, build a device form factor like what you see on the right represents the precision device, which has sort of echoes of other implantable electronics that many of us know well. Deep brain stimulators or the NeuroPace RNS device share a similar model, but in a different kind of bandwidth regime. That yellow electrode array on the brain surface is what I was showing you earlier. We have a head stage that sits in a recess within the skull, and then a tunneled subcutaneous connector that goes to a chest wall implant that's implanted in the chest wall that contains a wirelessly rechargeable battery and also wirelessly transfers the compressed data to a digital system. So here is a kind of a rendering that shows you in a little bit more detail how the system is configured. And I'll just give you a quick sort of sense of what we've been doing with these systems. I'll emphasize also, we're entering a world in which, these novel devices have to be manufactured in a reliable, quality assured way. And medical, the medical device supply chain is really not built around tiny features. And this is a new regime for medical devices and medical device supply chain to be working with things that are so small. Conventional electrodes that we know from the current standard of care are really macroscopic. And so moving to different spatial scales has required us to bring semiconductor manufacturing techniques into the world of medtech. That itself has been a technical and regulatory challenge, but I think it's incredibly exciting for not just neuroscience, but a number of branches of medical technology. And I just want to I'll show in a kind of an abbreviated way some of what we're doing in two different regimes, both in the motor and in the language restoration field. I think many of us have appreciated due to the work of a number of groups in the BrainGate and other consortia that speech output and fine motor control of the hands and limbs are really linked from a neurophysiologic standpoint. So a lot of the techniques that we've been able to apply to what we've learned over the decades about motor control, we can now apply to decoding, expressive language. And what we see, as you can kind of see in the video raster on the right, is that when we have an electrode array over primary motor cortex, and we've done this now a number of times in the context of awake DBS studies, in which we've been able to piggyback using our investigational capacity with collaborators at Penn, that fine motor control of the hand, fine motor gestures of the hand, each of those muscle synergies and each gesture, has a particular electrophysiologic signature as seen, on the primary motor cortex from our, high resolution array. And so many different hand gestures can be uniquely decoded. here I'll just show a couple, kind of like a rock, paper, scissors. And what you're seeing is time on the x-axis and the basically power envelope of the electrodes, in the gamma band on each electrode stacked on the x-axis. And what you can see just visually is that the time course of the intention to move and then the actual movement or the imagined movement of each of these actions is visually distinguishable and certainly at the level of a CNN or an RNN. Easily distinguishable, and that holds true for even at the level of complex individual finger movements. So we've done a fair amount of that, and in the context of awake craniotomies for language mapping in patients with tumors in eloquent areas, we've also been able to show that we can decode the intention to speak and the initiation and formation and imagination of individual words along the lines of, but not quite at the level of sophistication just yet, because we don't have the number of hours of continuous usage is what the UC Davis and other teams have shown, but nevertheless, consistent results in the regime of, speech decoding. So I think I'll just stop there. Some of our work is available on the bioRxiv in a preprint that will be updated very soon over the next couple of weeks, hopefully with the peer-reviewed version of the work. And certainly, I'm looking forward to the discussion here, and just want to say how much of an honor it is to be presenting with this group. And there's just such momentum in the field now. I think all of us are just feeling that this technology is going to make massive impact in medicine and in the world, over the next couple of years. And I can't wait to see this deployed as a standard of care and beyond.
Wonderful. Thanks so much, Ben, both for a great presentation and for showing those neural data, which I could look at all day and all night. It's always great to see those, and we can get a sense for just how powerful those neural data can be. And with that in mind, I will hand right over to now, not on the coast, but in the middle of the country, hand over to Matt Engel at Paradromics.
Perfect. Let me just pull up my slides really quickly. I have the unhappy task today of giving an engineering talk in a meeting with so much exciting clinical data, but I hope that I can make it at least a little bit exciting for all of you. Great. Can you all see my slides?
Yes, you're good.
Perfect. So I wanted to start off and talk a little bit about you've seen a lot of new hardware coming into the space. Brain-computer interfaces are changing. Hardware capabilities are changing. And I think sometimes the changes in devices and hardware can seem very esoteric and very technical. But I think that there's a really illustrative historical example when you look at the use of the internet and the evolution of the early internet, of how kind of boring hardware changes can accumulate into really transformative use cases. So for those of us who are old enough to remember using the internet in the 90s, it was largely text-based for a long time because you couldn't download images fast enough to have a visually rich internet. And it was only after enough people had enough download speed that you could download images very quickly that the internet started being a visually rich medium. And then, like, a great example is in 2010, Netflix switched from a company that sent you DVDs with fingerprints on it that you ordered online into a real-time video streaming service. And it wasn't that, like, the founders of Netflix had this genius idea to stream real-time video. It was that before 2010, not enough people had enough download speed to be able to do that. And what we're seeing right now in brain-computer interfaces is a different kind of evolution where suddenly we're getting better and better hardware. We're able to do some of the things in clinical trials, like communication, that have been done for a while. They're going to get better. And then there are going to be new applications that we haven't even thought of. And from my perspective, the most exciting thing about this revolution is that there are just many conditions, ALS being one of them, that currently there is not a cure. And we're all working very, very hard as scientists, as advocates, to try to bring about more biological discovery and bring about biological cures, but likely for a lot of people living with these conditions, they may not see a biological cure. But what we can provide them in the meantime are therapies that can really help change quality of life. And BCI can be that kind of therapy for across a large number of conditions. We can change really hard to treat medical conditions into solvable technology problems. And that's the mission of Paradromics. And that's something I think a lot of people on this call are very excited about. That in mind, we are building a high data rate implantable brain computer interface. We felt that, the three most important requirements for such an interface were having access to direct data, being able to read out the signals of individual neurons, to be able to read out from as many neurons as possible, because that in some sense determines the bandwidth of a brain computer interface in the same way that your download speed for home internet determines your ability to access the internet. Your ability to talk to a lot of neurons individually is ultimately the bottleneck for BCI applications. And then the other thing that we think is just really important is, in addition to just establishing this data link and trying to get as much data to be able to transfer between brains and computers as possible, we want to make sure that we're building a medical device that can perform on medical device timescales, i.e. 10, 20, 30 years without a significant decrease in reliability. Now, there's a little bit, I added this slide, I'm not going to lie, only about 10 minutes ago. There's a little bit of a thing that I want to clear up, just in defense of people like Lee and Romina and Sergey, who I think are not sociopaths, and they don't want to just poke little wires into people's brains for no reason. There are a lot of really interesting and orthogonal approaches to interfacing with the brain. But the reason why companies like Paradromics and Neuralink and BlackRock and research consortia like BrainGate are using probes that penetrate into the brain is because the sources of signal in these motor based tasks like cursor control, speech, robotic arm control are neurons that are located in deeper cortical layers. And in order to record from individual neurons, you have to be within roughly 1/10 of a millimeter from those neurons to distinguish it. And the quality of data that you get from recording from neurons versus recording on the surface of the brain isn't the same. It's not to say there wouldn't be great reasons to record on the surface of the brain, but any two individual neurons that you were to record from in the brain, if there is any measurable correlation between the two, it's very low, meaning the individual neurons are doing their own thing. And so the more neurons you record from, the more independent sources of data you have. When you're on the surface of the brain, if you bring two electrodes within about 1 millimeter of each other, they're more than 50% correlated. And if you bring them within, let's say, 1/10 of a millimeter, they're more than 95% correlated. So the physical analogy that's often used for this kind of situation is that you drop microphones into the bleachers of a stadium, you record individual voices. The same number of microphones into the parking lot, they're essentially recording the same roar of the crowd. Very correlated signals. So there are places for all kinds of different approaches, but it's not to say that penetrating electrodes are out of date. In fact, they're still the state of the art for getting data out of the brain at scale. We did, however, share some of the concerns about putting things deep in the brain and saw, much like Romina was describing, it's really important that you don't disrupt blood vessels as you're inserting things into the tissue. And there is a size dependence when you're putting something into the brain as to how it will interact with the brain, particularly the very small blood vessels of the brain. When you make things smaller than about 50 microns, you can think of it physically as like putting your finger through a woven blanket. It tends to do significantly less disruption to the blood vessel, which the network of blood vessels, which in return is very good because you don't release any kind of inflammatory agents into the brain. You don't recruit local glial cells and astrocytes, which are like the immune cells of the brain. And as a result, your brain is much gentler on the device over time. And I'm actually going to show some longevity data from our device that is very compelling, and as a result of making the electrodes so small. So we have a device with 421 tiny little microwires that reach down into the cortical layers and record signals. It's very, very similar to the device that Sergey described, this Utah array. It has a lot of advantages, however. We had the advantage of building this after looking at all of the success from the Utah array program, which Rob will tell you a little bit about later, I think. We saw one, people generally don't want things coming through their skin. They want everything to be completely implantable, completely concealed. Two, making the electrode smaller can increase the device longevity. Typically, this kind of single neuron signal that you get from implantable devices like this goes away after a certain period of time as the signal to noise gets worse. But if you can preserve a really good interaction with tissue, you can continue to measure signal neuron signals for a long time afterward. We also have a wireless power and data unit. It's inductively powered, so it doesn't have a battery, and it's continuously powered by a wireless link. And it has an infrared data source that sends data out through the skin to an external transceiver. And so the data link can be run at about 100 megabits per second, which supports more than just one cortical module. In fact, it supports more than 4 cortical modules. The whole backbone of the system would support in excess of 10 cortical modules running simultaneously. But our current system is spec'd for 4 of them. And so in total, this is 1,684 intracortical electrodes. It represents the highest bandwidth BCI system in the sense that it has a lot of channels and can record from single neurons. We have had devices now implanted in sheep for over 2 years, and we've been measuring the single neuron signals from those sheep over that time period. And what is very exciting, especially probably to some of the people here on the call who have been involved in intracortical work, is that the signal to noise ratio over that time really hasn't changed very much. And if we extrapolate the changes in signal, we would say that this device will still be recording from individual neurons more than a decade from now. So the device performance will outlive the sheep. Our sheep don't speak, unfortunately. We haven't trained them yet. And so it's hard to compare on a performance basis between the sheep and other like cursor control or speech reconstruction tasks. But we can talk about bits per second in our sheep decoding. We present the sheep with auditory stimuli, and then we decode what auditory stimuli were played to the sheep. And based on the error rate and the complexity of the sounds we're playing, we can back out a bits per second. And so right now we have sheep that we're decoding brain activity from the sheep at 94 bits per second. To my knowledge, that's the highest recorded BCI decoding data rate. I'd be happy if anyone on this call knows of a higher data rate. Please chime in. Okay. Perhaps the part that's most germane to this discussion, we are getting ready to go into our clinical trial for this system next year. It'll be an early feasibility study. We'll be using this device to restore speech and cursor control to patients with conditions like ALS and high spinal cord injury. You've seen already the kinds of really impressive things that an intracortical approach with many channels can do in terms of speech decoding. Our system will have twice as much data as the system that Sergey showed you earlier. So we would only expect higher communication rates with lower error rate. We're very excited about that. And some of the people on this call will be working with us on that project. We're also very excited about combining that with cursor control, because if you can do effectively voice to text and fluid cursor control, then you have the ability to control a computer at the level of a person who is able-bodied. And so we want to restore people, at least in the digital domain, to a place where they were before their diagnosis or before their injury. A little bit of traction so far with the FDA. We've had 2 breakthrough device designations. We're part of the TAP program, which means we get to have a lot of meetings with them in advance of our IDE submission. And we're getting ready for our first in human experiments in the second half of next year. So thank you very much for listening to this talk. I know it was engineering, no human data, which would have been really exciting, but I'm looking forward to seeing you next year when we can talk to you about the results of our early feasibility study. And then I also just wanted to leave everyone with this quote, which I personally think could be the mission statement of the entire neurotechnology field.
Awesome. Thank you very much, Matt. It was a great update and good to see the great progress being made. We will now turn to moving, I've lost track of which direction we're going. We're certainly headed west, though, assuming that Rob Franklin at the moment is in Salt Lake City. We heard a little bit about the Utah Array a little earlier from Sergey, and we'll hear now a little bit more from Rob at BlackRock.
Thank you so much, Lee. Give me a minute while I get my presentation up. Okay.
You're good.
Okay. Perfect. So I'm super excited to be talking with this group today. I've known several people, including close friends, that have experienced ALS and the symptoms of ALS in their life, and excited to be doing whatever I can to help this community. I'm a child of the 70s and 80s, and so, Matt, I definitely understand the low data rates that we experienced in the 90s and how that works. But I also, in the 80s, as most kids who grew up in the 80s did, I was a fan of Star Wars. And from the moment I saw Luke Skywalker getting a bionic hand that functioned almost as well as his own biological hand, I've been hooked on bringing that idea to reality. And so for the past 15 years, I've had the privilege of working for BlackRock Neurotech, leading the way forward and bringing function back to people across the spectrum of human ability. When I first started, the Utah Array was the primary technology that was being used for brain-computer interfaces. Today, there's a variety of different technologies that we've heard about today and some that we haven't, but it's super exciting to see the technologies that are being developed today, where those are at, and what's going to be coming again in the future. BCI is being heavily influenced, at least at BlackRock. We're looking at how it works with machine learning and artificial intelligence. These are areas that seem relatively new, but we've been using machine learning for many, many years, almost since the first implant, to decode the signals that are coming out of the brain. And with the advent of the generative AI solutions that have been coming out, I think we're going to see some really cool applications where all of these are getting applied at the same time to really increase the amount of data and the amount of control people can have around the world around them. So in the next 5 years, BlackRock is working on higher density, longer lasting implants. We're working on improving the user experience in a number of different ways and making sure that we place users first. That's been one of our hallmarks, is we've always tried to think about the user experience and place the patients first in what we're doing. And then we expect that we're going to start seeing initial systems with regulatory approval. We saw Neuralink and the Synchron system's already quite far along in that pathway. I think Matt and his group with Paradromics and Ben with Precision are also going to be pushing theirs forward, and we may see them getting approval in the next 5 years. And then we also expect to start seeing exploration of new applications, maybe vision, rehabilitation, some different areas that haven't been explored. The speech things that Sergey presented today are actually fairly new and fairly new to the experience at BCI. So I think there's going to be a lot more of those types of applications that we see as we start to explore what is possible and where BCI can go. As we do those explorations and push forward, it's important that we continue to be collaborative and working together as a community. Lee has put together the IBC-ICC, which has been a fantastic place for people to work together and come together to discuss in the pre-competitive space how we can improve BCI for everyone and make sure it's the best. We need to make sure that privacy is first and foremost, that people's neural data is their neural data and it's not being sold to other third parties or being used in ways that haven't been given permission for it. And then above all, that there's ethical oversight, that we're constantly thinking about how do we make sure that we do brain-computer interfaces in an ethical way that is going to advance humanity and advance the entire society and community. So with that, I'm going to close out just talking about the patients that we've worked with. I've had the privilege of working with most of these people. It really is why we do what we do, because of seeing the benefit and the joy. Every time I watch that video, Sergey, of the first time you hooked your patient up and seeing the immense happiness and joy from hearing his own voice again, like that's why we're in this field and why we're pushing BCI forward, so that we can really enable people to be their best selves and be who they are. I'm going to close with the story of James Johnson, who's one of the BCI research participants I've had the privilege of working with out at Caltech. And he's going to talk a little bit about what does BCI mean for him and why is it important for him to have a brain-computer interface.
All right, James, you ready?
I'm all set. And I came across this program where your brain can interface with a computer.
I'm just going to do a quick test.
I didn't even hesitate. I said, sign me up.
All right, so what we have here is our view into James's brain. Each one of those neurons is telling us something about what James wants or experiences in the world. And we can use that to train statistical models and allow his brain activity to drive motion of the cursor.
I'm imagining following that cursor, kind of like a PlayStation controller. To think that I can control the computer, the cursor, just by thinking about it is mind-blowing. But now I'm going to apply it to what I love to do the most, Photoshop. So right now I'm just going to do a simple blend of 3 different photos. We're going to put her face in the background, get like a soft brush that allows me to fade and make my brush a little bigger. I'm just gonna fade.
We need to create a new Bob Ross series with you in Photoshop.
Art is something that I can do that belongs to me. It gives me control over something that's just me. I don't need assistance. I don't need someone to hold the spoon and feed me. Art allows me to have an identity again. Well, BlackRock's technology has given me hope. It gave me hope that someday I will be able to interface with not just the computer, but my environment. So the sky's the limit with things that I could create.
And with that, I will wrap up and be happy to take any questions in the question and answer session at the end. Thank you for your time.
Thank you, Rob. Thanks for the great update and summary of what's going on with the BlackRock systems. I will now, you've heard a couple things about this before. I'm going to turn over to Jessica Kellerman, who I believe is sitting in Boston, who's going to tell us a bit about the implantable brain-computer interface collaborative community. Jessica, it's all yours.
All right, great, thanks. Let me share my screen. Right, you can see everything okay?
Yeah, you're good.
Great. So hi, everyone. I'm Jessica Kellerman, and I am honored to be here with you all. I am the program manager of the Implantable Brain-Computer Interface Collaborative Community, or the IBCICC. And my work centers on bringing people together to drive innovation in IBCIs through structured collaboration. And today I'm here to talk a bit more about the IBCICC, what we do, why it matters, and how you can be a part of it. So this is the agenda. I think it reflects those goals pretty well. So the first thing I'll do is introduce you to what the IBCICC is, and then we can discuss how the IBCICC can support the ALS community and vice versa. And then I'll talk about the role of the IBCICC in the IBCI field, and then we can go over how you can get involved. So the IBCICC is a collaborative community made up of a wide range of stakeholders, including researchers, clinicians, regulatory experts, industry leaders, patient advocates, and most importantly, people with lived experience of paralysis and their care partners. We currently have over 300 members who are all committed to advancing the development, safety and efficacy evaluation, and wide and equitable access to IBCI technologies. And we are the first FDA participating collaborative community in clinical neuroscience. And we're so fortunate to be able to gain and share perspectives with the incredible people at FDA's Neurologic Devices Office. And I'd also like to acknowledge EverythingALS as one of our signatories, and especially thank Indu Navar for her critical role in helping establish the IBCICC. Our collaboration with Indu and EverythingALS really reflects our commitment to draw from wide perspectives in the ALS community, and we hope that many of you here today will consider joining this effort by contributing your experience. Whether you're a person with lived experience of ALS, a care partner, a clinician, or another stakeholder in the field, we really value your input. The IBCICC's work will help to address a wide range of challenges toward bringing safe, effective communication technologies to people with ALS. So you've just heard from companies and academic researchers developing IBCIs. And ordinarily, when these entities interact with federal regulators, those discussions happen properly and for good reason in confidential silos. The IBCICC provides for open, transparent discussion across stakeholders in a carefully constructed pre-competitive space. This unique forum was created to reach well-informed consensus across a range of key issues. Your insights, those of people with ALS, care partners, and advocates, are crucial to shaping the future of this field. So now that I've given a bit of an overview, I'd like to explain the role of the IBCICC in the IBCI field and how we are structured to address problems in the field. IBCIs have a long academic history dating back to the 1960s with motor cortex decoding in monkeys, but it is really within the last few years that we have begun the steps for commercial translation and getting IBCIs to the people who need them. Embarking on this process has raised a host of critical unanswered questions. Examples of these questions include, what are the outcome measures that will lead to product approval and reimbursement? What guidelines should clinicians follow? What are the best practices for handling neural data? These are not questions that can be answered in silos. Progress is dependent on the IBCI field collaboratively answering these questions. And the IBCICC aims to do that through topic-specific work groups. We have 7 work groups, each addressing a unique aspect of IBCI development. While all of these have relevance to the ALS community, I'll highlight just a few that may be of interest. So the first one I'd like to highlight is user priorities/preferences and IBCI use cases. And this work group focuses on understanding and incorporating the perspectives of potential users into the design, testing, and application of IBCIs. I'd also like to highlight the clinical study endpoints work group, which is driven by the need to clearly define measures that can be used to obtain regulatory approval of IBCIs. And finally, I will also highlight the clinical practice guidelines work group, which is working to establish evidence-based guidelines to ensure safe and consistent care for individuals using IBCIs. So over the past 8 months, we have been building momentum in bringing IBCI stakeholders together through these work groups. We launched the IBCICC and our 7 work groups back in March and April. And since then, we've had over 330 members join, and we've held 27 work group meetings. And we also had our first hybrid annual meeting in September. And over the course of this time, the work groups have begun working on a number of outcome-oriented projects. So just to name a couple of specific initiatives, the user priorities work group is actively developing life journeys of potential users to better educate medical device manufacturers and researchers, and to better inform a common understanding of the clinical trial outcomes that may be important for individuals with ALS. Another example is that the clinical practice guidelines work group is seeking to identify the range of clinicians and how they will identify, refer, and support IBCI users with ALS. All work group efforts are really geared toward reaching consensus on major issues in the IBCI field. And we would really like to have your input as we drive toward consensus. So I would like to invite you all to become a part of the IBCICC. By joining the IBCICC, you can contribute your expertise, share your insights, and help us guide the direction of the IBCI field. You will not only be supporting the growth of life-changing technology, you're helping us shape a future where individuals with ALS and other conditions have new, meaningful ways to communicate and connect. So we welcome you to be involved in your able capacity. You can be an active participant in our regular meetings, contribute to offline activities, such as collaborating on files or providing feedback, or you are also welcome to join in once per quarter to provide your input. We are very flexible, so please only contribute as you are able. If you'd like to get involved, please visit our website at ibci-cc.org and click the join button. Together, I truly believe we can make a difference. And with that, I would like to thank you for your time, and I look forward to your questions during the Q&A session.
Excellent. Thanks very much, Jessica. And I'd like to thank all of our speakers today for what was really a wonderful overview of the progress that's been being made in implantable BCIs. I'm going to take just a moment. I'm going to ask Christian Rubio, you're out there. You've got the stopwatch. And I know we started a little bit late and may have slipped just a bit. It is up to you to tell me whether we should have a brief Q&A or whether we should strive to get back on the symposium's clock. Or Jen, either Jen or Chris, who's ever jumping in.
Jen, I'm going to
I'd say let's go for it. I think this is all such great talk, and as long as everybody wants to stay on and you're good with it, I think questions are always great if people have them.
Yeah.
Okay. All right. Excellent. So we'll take the next 2 hours for question and answer. And all right. Let me, there was one kind of broad topic that a few people touched on, both in the Q&A and also in their presentations. And I'll let anybody jump in from our speaker group that would like to. There's an open question about the importance of ethics and particularly of how we handle these neural data. And a few people made some comments about it, but if there's anybody that would like to kind of speak to this about either how they're using neural data or how they're thinking about the sharing or not sharing of neural data right now, and just wants to comment a bit, please just jump right in.
I can make a comment. We're currently working with across a number of academic centers with a variety of collaborators, and thinking very hard about how to make some of those data sets more widely available beyond beyond their initial use cases, which I think also will be multi-purpose. So I think it's really important to drive the field forward. It's nuanced, as all of us know, especially those of us who are who are working on the commercial side. But at the same time, I think we learned from a number of technological predecessors, including I think from the world of genomics and digital pathology, that there's so many things that can happen when you try to democratize the data a little bit, and use cases emerge and accelerants emerge beyond what might initially have been imagined. So we certainly on the precision side are working to try to share data in certain ways, and I hope we'll be able to do that across the industry in some capacity.
And I'll add maybe, Lee, just my 2 cents on that topic. That I think there's an element of sharing. Of course, I think all participants in the trials electively say, yeah, you can use my data. So that's one point. And I think we're at a point now where there are some elements of collaboration that make a lot of sense to say, hey, how do we look at this data all at once? But then there's, I think, also when you think about that commercialization element is where, just like everybody walks around today with a phone and there's privacy to that, right? So I think we need to adhere to the same rules and responsibilities as regards to privacy of information. And so even when we're looking at clinical trials, there's going to be a segmentation of intent data that we can see. But if they're interacting with the digital device to do their, say, activities of daily living, that information is something that we actually don't have and may not have preview to that. So it'll be an interesting balance as we move forward with these technologies. What data can we see? What data can't we see? So yeah, I just wanted to add my 2 cents there.
Great. Thanks, Kurt. Romina, did you have a?
Sure, yeah, I'll comment on this. Yeah, so at Neuralink, we very much value transparency. And so, as Kurt mentioned, we do have informed consent for all of our trial participants, where we present in exact language what we do with your data, who your data is shared with, in layman terms. So it's not like your iPhone privacy policy statement that's like 30 pages long and no one actually reads it. We present it in very concise, clear terms for all of our participants, and they have the options of opting in or opting out. And so I just wanted to specify that from the PRIME study.
Great. Thanks, Romina. That was a good, that discussion, of course, about sharing and neural privacy is one that's really important to the field and will continue. And I think that we just heard some great and really important perspectives. There's one question in the chat that just came in from Dr. Ostro. I'll read it aloud since not everybody maybe see the chat. Progress in BCI is just amazing. People living with ALS often have the same question: when could we reasonably expect this incredible technology to truly become part of accessible care for people living with ALS? And what are the deliverables, goals that have to be achieved to reach this point? I'll give a brief answer to both. The when is I hope as soon as a pivotal trial is complete. Just for, and not everybody on the call may know the path that device trials take. They start as a pilot trial, and then they have a somewhat larger pivotal trial. And it's the end of that pivotal trial that most commonly there's a regulatory decision about whether that device may get its market approval. And I'll let any of the companies who, of course, are the ones that will create or are creating the final form of medical devices kind of give their independent predictions of when that time may occur. The deliverables, the second part of this question, is one that everybody is focused on, because for anybody with ALS on this call, any of their care partners of people with ALS, I suspect that there would be a range of answers to the question, what would you like to do with the technology that you just saw? And all of those answers are good. And somehow we'd like as many of those answers to be considered a deliverable or a goal, so that as these devices work their way as quickly as possible, demonstrating safe, effective devices, that those devices will ultimately be able to satisfy the needs and the hopes of the people for whom these devices are being developed. So the deliverables and goals are really going to be, I think, part of an iterative discussion. The collaborative community is one place to have those discussions. But I think we as a field would like to hear repeatedly, what would you like this device to do? How would you like it to help you? And I think everybody on the call involved in either developing or testing these technologies wants to hear those answers over and over, because it really does shape in real time how the devices are developed, how the clinical trials need to be designed, and ultimately what those outcome measures are. That was a long answer. Anybody want to add or disagree?
No, I will certainly agree, Lee, from my standpoint. I think from a delivery, like a time frame, I think you saw a lot of great technologies from the implantable BCIs, and totally agree with Lee that this is going to take a partnership. Well, I think as a field, but also, of course, with the FDA, right? These things need to be safe. They have to be shown to be safe over a period of time, and they also need to have a effectiveness endpoint, and there's that balance, right, that you have to meet within that pivotal trial. I think as you look at the next 3 to 5 years, I think that's going to be a window where you start to see these technologies become potentially available. But I do want to stress one thing: like these technologies don't become available without participation. I think we have some really amazing, really pioneers. From early on with the BlackRock system, the Utah Array, and to all the way to today, where people are participating in studies. And without those pioneers, without those folks participating, like these technologies don't evolve, because it ultimately needs to be in these early studies and then into a pivotal study. So, thank you, I guess, all for being, those that are the pals online to just even start to learn and educate yourselves around what's going on in the field. So, yeah, just wanted to add that, Lee.
Great. Thanks, Kurt. Rob, I saw you raise your hand.
Yeah, I was just going to say, I mean, I think the easy answer to the question is that they will be available as soon as we can present to the FDA data that they are safe and effective for clinical use. And the FDA really safety, I think, a lot of the groups have a pretty good head around, but what the FDA is going to accept in terms of effective use is still an open question that many of us are actively discussing with the FDA and also as part of the IBCICC. In terms of like what it can enable and what it will do, I will say that for me, I will not be happy until someone with a BCI can interact with the computer and the digital world around them at the same speed and with the same effectiveness that I can do it. Or most of the people in this group can do. So I'm going to continue pushing to advance the speed and how fast we're getting data in and out of the brain until we can get to the point that it matches what an able-bodied person can do.
Thank you, Rob. And I think with that hopeful phrase and conclusion, I know that there's a lot more questions. I saw a few in the chat and elsewhere. But in the interest of moving things forward, I saw Jen look at me and I saw Christian unmute, and I'm going to take both of those as a cue to wrap today's session. I will just say that you heard from a number of people today who really are dedicating every waking hour to developing brain-computer interfaces to help people with paralysis. And as a few just alluded to, it's only with the enrollment in the current and future clinical trials of people with ALS and people with other forms of paralysis that we can learn how these devices work, how to make them better as quickly as possible, and to get them across the finish line to safe, effective devices that are not only approved, but can be reimbursed through our healthcare system and can be equitably and widely distributed. We all look forward to that day. We look forward to your help in the collaborative community, in the clinical trials and elsewhere. And I just want to thank both Everything ALS and ALS ONE for the opportunity to host this session today. And I will hand back to the two of you.
Thank you so much. I just want to say a few words before handing it over to Christian. This has been just so incredible. Thank you so much, Lee. Just a huge thank you to you and to Indu and everyone at Everything ALS for this truly inspiring day. What a wonderful way it has been to wrap up our 3-day annual symposium. So grateful To all of today's speakers and moderators, thank you all so very much for sharing your time, your important work, your thoughts, ideas, and collective vision with each other. Just a profound show of collaboration. Some would say that, geez, no one shares anything. And I think today was just clearly stated that there's a lot of collaboration, there's a lot of sharing, and there were no egos on display today, and your hearts were glowing so brightly. It was just very, very cool to see, just amazing. As for the last 2 days, the research that was shared from pharma and academia brought so much hope in the form of promising treatments in the works to slow, stop, and ultimately cure the disease. And what was shared today, showing what is already happening to help that science advance more quickly through technology, and to very importantly, probably most importantly, show how technology can, and as shown in so many inspiring videos, how it's already improving the quality of life for so many battling ALS and so many other diseases and disorders, and through trauma right now where people are. And it's just been overwhelming and inspiring. I'm happy to report that all of the sessions today and last 2 days were all recorded, and they will be sent out to all attendees who have registered. Remarkably, there have been 780 people who have registered, who have sort of filtered in and out over the past 3 days from 32 countries. Just incredible. So those recordings will be put on the symposium website when they are ready, and they will be sent to all registered attendees as well. So stay tuned for those. And just give me a little time to extract them and get them ready to go out. But I wish everyone a great weekend and a really special holiday season. And with that, I will let Christian close us out. Christian, thank you and Everything ALS so much for spearheading this important day to wrap up our symposium.
Thank you so much. Thank you, everybody. Lee, thank you and all the presenters this afternoon. Thank you for showing us the state of the now and also what's next. Brings so much hope to the whole community. And also thank you for everybody from the morning session as well, from James Berry and Lyle Ostro to all of the presenters and discussion leaders who shared so much about their opinions and their thoughts on how we are going to continue to innovate with open innovation and open data to accelerate and just be brave with each other and collaborate directly with each other. Speaking of working as a community, one next thing that's really important next week is the advisory committee meeting with the FDA on digital health technologies. That's happening on Wednesday and Thursday. There's a live stream for that. So as a community, I invite that we all be present for that if we can. It affects all of our work to make sure that they understand the critical value of the work we're doing here, and then the considerations that everybody discussed today are on top of mind for us, and we are the ones to inform them. Other than that, as well, we have our December roundtable. Jen, we'll be working with you, of course, and as we develop our agenda for that and share that. And I'll close out with simply that this is one of the great partnerships we have. ALS ONE is an amazing organization in its own right, run by Jen and her team. And we thank so much the work you do, not only to support the education and the awareness that you bring, but the fundraising you do as well for so many clinics like MGH and others that benefit from it. And ultimately, you live and represent the experience of our community. And it comes back to every single person's effort and contributions that they make, whether it's participating in studies or simply just fighting the good fight with us wherever they have the energy and the kind of gift to contribute. So we value each and every one of you. With that, thank you again. Hope you enjoyed today's program as part of the symposium, and we can't wait to do this again.
Thank you all so much. Have a great day. Thank you, Christian. Thanks, everyone. Thanks, Lee.
Thanks, Jen. Thanks, Lee.